and the yellow bile and so on, and the mucus. Others, they say, no, there must be a solid abnormality in order to have a disease. Others, they come up with a cell and then molecular aspect. So nowadays, the modern pathology, the way we understand diseases nowadays is that there is always some molecular or biochemical changes whenever there is a disease. So as you know, our body so is composed of many systems, for example, digestive system, reproductive system, and so on. And each system is composed of organs, and each organ is having different tissues. And each tissue, of course, is composed by a kind of cells. So to understand the tumors, we must understand that each tumor will arise from a cell. There is always a cell at the origin of any tumor, being a cancer or a non-cancer tumor. And as we shall see for cancers, there is always molecular changes. There are mutations or abnormalities within our genome in order to have a cancer. So that's why we will refer most of the time to the molecular biology you had in order to understand these things. So a cell, as you know, is composed of, I can say, three main parts. You have the plasma membrane or cell membrane, which is just like the outer envelope. Then we have the cytoplasm with many organelles such as the Golgi apparatus, which is Golgi complex. So we have the mitochondria, we have reticulum, endoplasmic reticulum, which can be smooth or rough, and so on. And we have the nucleus. So nucleus, cytoplasm, and the cellular membrane. So pathology, last time I had this definition, is the science of disease or suffering. So the objective for pathology is to identify and describe the different parts of a disease process as we have seen. And you have seen the four parts of a disease process, which are the cause or origins. And sometimes the cause you don't know, but there are factors. So yeah, if you said that drinking alcohol is a risk factor to have a cancer of the liver, it is not a direct cause, but it is a risk factor, because not everybody who drinks alcohol will have hepatocellular carcinoma. So there are mechanisms then. That cause or that factor, the way it works in order to produce a disease, that is mechanism or pathophysiology. Then there are structural and functional changes, which are characteristic of the disease. So structural, you can understand if there is a swelling or there is atrophy, decrease in the size of an organ, or the organ becomes larger, there is ulceration, many things, hemorrhage. Then functional changes, can see it at different levels. So it can be changes at the function of an organ. For example, a liver is no longer producing albumin and other hormones. And also the limb. If a limb is having a cancer, it will not you will not work. So that is a functional change at the physical aspect, but also functional changes at the molecular aspect. And the consequences or results of a disease. So this we have seen last time, the history of pathology, humoral pathology, solid pathology, cellular pathology, and molecular pathology. So what is a tumor then? So tumor itself, this word, when it is in Latin, so it will just mean swelling. And this we have seen in inflammation, that in the 5 cardinal signs of inflammation, we have one called the tumor, which just means swelling. And this has not to be confused with the tumor as a mass itself, a cancer and benign tumors. So tumor, when it is written in the sense of inflammation, is not the same as the tumor when we come to neoplasia. But when we are in tumor pathology or neoplasia, tumor will mean usually new growth or new mass. So that's why you will be very careful when you are describing what you see. If a patient comes with a swelling, as a medical doctor or as a dental surgeon, will not say this is a tumor. No, it is a swelling until you have all evidences that this is a true neoplasia or tumor. So therefore, the term tumor can be applied to any swelling. But in this chapter, when we talk about tumor, it will refer to neoplasia. So neoplasia, which is the Latin word, means new growth. So in the book of Robbins, as I said, it is the conventional book used to teach pathology all over the world, where they speak English. So neoplasia, it means new growth. It is the abnormality of cellular differentiation, maturation, and the control of growth that results in the formation of masses of abnormal tissue. And these masses of abnormal tissues, they are the one we call tumors. So tumors, in this chapter, we refer to the masses of abnormal tissues. So the difference between neoplasia and just a swelling, for example, inflammation, is that neoplasia will persist even if you remove the stimuli. So in inflammation, when the cause was, for example, bacteria or a foreign body, when you remove it, the swelling will go. But for tumors, neoplasia, if there is a cancer which was caused by let's say a bacteria, Helicobacter pylori, you can treat that bacteria using the most strong antibiotics and so on we use in this country or over the world. But if the tumor is already there, it will not reverse, it will persist. So it's a risk factor. If removed yet, it is removed yet, the mass persists. So this is a sign that it is not just a swelling, it is a true tumor. So the causes are both genetic and environmental, and they play roles. So you cannot say a cancer is caused by only environmental factors or only genetic factors. They usually work together. And some people in literature, you will see that they say for tumors, 60% of the risk of having tumors are caused by environmental factors, while 40% come from genetics. And this has an implication. If 60% of the risk of having a cancer is caused by environmental factors, it means these are things we can control. And what I was saying by implication is that there are things we can do in order to prevent such cancers. Of course, we cannot change our genetic makeup, but we can work in a way that that genetic makeup will not be influenced by the environment to cause cancers. And this is why you will have always to educate the population, your colleagues, students in other domains, in your churches and so on, in the community, to educate them about preventing cancers. And this is why, in our country, they give vaccination against a virus which causes cancer of the cervix. That is to act on the environment in order to limit the occurrence of cervical cancers. So that's why we can do all possible or things possible for us as humans to limit the occurrence of cancers, but the cancers will not disappear completely because there are factors we cannot control. But at least we can control those in environment. So we have two types of growth or disorders, non-neoplastic. This means it doesn't cause a neoplasia. It is not a neoplasia. It is just a secondary to other causes. For example, inflammation. Here, the good example is inflammation. someone got a bite by bee or mosquitoes and so on, they will have a swelling. That swelling, it is not neoplastic, it is just a reaction to the bite. Or someone has been bitten or traumatized and so on. So neoplastic one, these are the primary growth abnormalities, as we shall see. So the difference, as I said when we started, the neoplastic proliferation or the tumors which are neoplastic, swelling which is neoplastic, it is once it is there, even if you can remove its cause, that mass will persist. While for the reactive ones, non-neoplastic ones, once you remove the stimulus or the cause, the swelling will disappear. Another thing is that neoplasms are uncontrolled and are irreversible. So irreversibility is the same as what I was explaining. You remove the cause, the mass persists. Uncontrolled, it means our body is unable to continue to control things. And we have two types of this neoplastic proliferation. Benign tumors. These are localized. They do not invade or infiltrate. They do not destroy surrounding tissues. So usually these are not, they don't cause death. But the malignant one, malignant tumor, it is the same as cancer. So these are invasive. They destroy, they infiltrate, they spread wherever they want. So, and you can understand that they cause death. They cause death. So benign tumors, you will see many people having the tumors, they grow very slowly, they don't destroy surrounding tissues. But of course, there are exceptions whereby these tumors they can kill. But these are very few exceptions. Otherwise, you can just remain with them throughout the life without causing any other problems. Except disfigurement, if it is on the face or somewhere where it is visible, it can be, it is just a matter of aesthetics. Otherwise, usually not threatening the life. So the mechanisms of growth factor, these are the diagrams to explain. And here you will understand the third difference between neoplastic and non-neoplastic growth disorders. Here we have a normal tissue. It has here represented by two types of cell. One cell is yellow and another type of cell is red. So it means a normal tissue, normally or organ is having different cells, epithelial cells, fibroblasts, smooth muscle cells, skeletal muscle, and so on. So when it is a matter of adaptation, these cells, they can increase either in size, and you have run this in the very first chapter of general pathology. When it is increased in size, you call it hypertrophy. And when the cells increase also in number, you call it hyperplasia. So, for example, here we have both. You see that the size of a single cell has increased, but also the number of cells have increased. And this has happened to both types of cells, the red and the yellow ones. So this is an adaptation. It is a sign of adaptation. And it is like a combination of hyperplasia and hypertrophy. So when, and this is polychronal. So this is what I want to say by saying that there is another difference we shall see. The adaptative swellings, they are polychronal. It means, or the cells in that area will have the same features. If it is hypertrophy or hyperplasia, or will go. For benign tumors and malignant tumors, which are cancers, usually it is only one cell. So you'll find in the stomach you have many cells, but you will find only epithelial cells have undergone changes. Other cells remain the norm. So this means only one clone or one type of cell has undergone changes. So when you see this, it is very alarming. So for benign tumors, so you see that it is monoclonal, arises from only one single type of cell. So the cells, of course, they have increased in size, they have increased in number, but it is only one type of cell. So the yellow ones here, they have not been involved in this change. And usually they have a membrane around them. We call it capsule. Capsule. So n'agasashe kabitwikira. So the benign tumors are limited by a capsule. So all of these things I am explaining, they help you to clinically to differentiate benign and malignant tumors. So as doctors, this is a basic thing you will do. If you receive a patient, you must know how to describe. Is this tumor well circumscribed, having a capsule or not? If it has a capsule, most likely it is benign. If no capsule, if it infiltrates, you will keep in mind that this may be a cancer. And then you will have to discuss with your seniors and then again they give a diagnosis. And sometimes you will be alone to decide. And if you think you are in a district hospital, maybe you are the most senior medical doctor or dental surgeon there. So you have to decide according to what we are describing and then say, so because of this, a tumor which is not well circumscribed, no capsule, it infiltrates, it causes hemorrhage and necrosis, and it grows very, very quickly, very rapidly. So all these will be signs or symptoms that this may be a cancer. Then you can refer that patient to a referral hospital confidently. But we see sometimes the people, they just send, we think this is a tumor, and they don't make an effort to say, to describe what they saw. Because when you describe things also, you are learning. But when you work as like someone who is not a health professional, usually you backslide. So your knowledge goes atrophying, if you can say so. So this is a benign. So how about the cancers? Cancers, you see, it is also the cells are of one kind of cell. So one type of cell. But you see that usually here we have something I have not explained. This is like a basement membrane, you know, in histology. For example, below the epidermis, we have a basement membrane separating the epidermis from dermis. So when it is a benign tumor arising from the epidermis, it will not destroy that basement membrane. But when it is a cancer, it destroys that basement membrane because it is infiltrative. So you see here that some cells have destroyed the basement membrane and have gone in deep. So, and if it has a capsule in the starting, at the end, the capsule will be also destroyed. So here there is a yellow line here, which is just is representing a capsule. So malignant tumors are monoclonal, arising from one type of cell, but the cells, you see that the cells are not of the same size or shape. So you see, this cell is smaller than this one, and its membrane is irregular as compared to other cells. So even if it's pleomorphism, pleo means many. For those who know Latin or Greek. So pleo means many, morph means forms, form in France or shape in English. So it means the cells that they have different shapes, size, and so on. So this sign we call in histology or histopathology, we call it pleomorphism. So here we are showing now a pathogenesis as an example of lung cancer caused by smoking. So I think everybody knows that cigarette smoking is a very big risk factor to develop lung cancer, but there are many other cancers associated with it. But the lung cancer is the most common one. So this is the normal epithelium. So you know that in the respiratory tract, we have the epithelium there. It is columnar and ciliated. This cilia, they help to remove maybe dust, to move dust, because when we inhalate the dust or other materials. And also, and you have gallbladder cells which secrete mucus. So here they are not shown. So I think this you have seen in the first chapter about the adaptation, where you saw hypertrophy, hyperplasia, atrophy, metaplasia, and dysplasia. So metaplasia comes when? It comes when the body cannot withstand or resist the environment. So for example here, when someone is smoking, when someone smokes, these simple columnar epithelial cells of the respiratory tree will not resist or will not withstand the smoking. So for them to withstand, they have to change their nature. So to change their nature, but changing from one type of mature tissue to another mature type of tissue, it is called the metaplasia. So here they change from columnar ciliated epithelium to stratified squamous epithelium. Stratified because we have many layers, and squamous because the top cells here they are flat. So this is the first change. So when somebody continues to smoke, so what happens? These cells also they will say, no, we cannot resist again. So we have to adapt ourselves. This adaptation, the third one is dysplasia. So dysplasia usually when we see it, it is a sign that very soon a cancer will start. So it is a very dangerous disease. So in this metaplasia, we see it in many tissues, including the cervix. So what I am describing here, it is almost the same as in the cervix. Cervix, we have the endocervix and the exocervix. Endocervix is lined by a columnar epithelium, not ciliated, of course. The ectocervix is lined by squamous epithelium. And where they meet, we call it junction. In this junction, it is usually where human papillomavirus works to make these cells change their character. So these cells which are usually columnar, they will be changed into squamous. Okay, wambaza uti none se ko aho squamous na columnar biba bituranye mwe mu bibari twari metaplasia. So that would be a good question. Tubirebera ko ari metaplasia nuko dufitemo na gland. There are glands in the cervix. So these glands are columnar cells. They are never squamous. So when you see that these glands, some of them they have become they have this kind of tissue, a squamous structure of squamous epithelium. We confirm there is a metaplasia. So this is not a big issue. But it is a sign that something is going on. So as a health care professionals, you have to act. So when you don't act, then after some months or years, you will find it is dysplasia. So dysplasia is a changes kwabonye hejuru. We have, for example, here lack of maturation. You see that for here the cells on the periphery here on the surface they are flat. And their nuclei, usually they are not seen, they are very small. So when there is dysplasia, the cells on the surface, they appear the same as the cells on the basement membrane, basal cells. So this is a bad sign that changes have happened. So if you don't act, problems will come. So as medical doctors, because of course, dental surgeon will not treat gynecological diseases. If you have a woman, a lady comes to you just for screening, so baraza cyane sashka turabona hafi buri munsi baraza bashaka kureba ko nta kibazo bafite kuri service. So hari ibintu bibanza gukorwa ngira welcome sana mu Kinyarwanda. I hope everybody understand Kinyarwanda. So I said the junction between ectocervix and the endocervix, it is mainly aligned by columnar cells. So izi columnar cells ziba zifitemo glycogen n'ibindi bitandukanye. Mwarabyize muri shimi no muri secondaire ko iyo ufashe glycogen ugateranyaho iode hari kurera bitanga ijya gutukura cyangwa brown. So bigenda bitaho kwa muganga test bakora ya mbere hari icyo bita colposcopy ni icyuma winjiza muri genital organs, then ukabureba cervix neza desire haba hanamurika baba bamuritse n'itara. so iyo usizeho ikintu gifite iodine iode za cells zikaba ziri normal ziba brown. Iyo za cells zitakiri columnar cells no longer having enough glycogen, aho kugira ngo zibe brown zihinduka umweru. So iyo zihindutse umweru ni ukuvuga ko ahantu habaye either metaplasia cyangwa dysplasia. Icyo gihe ntabwo kubitandukanya kuri colposcopy bishoboka, ahubwo icyo gihe nk'umuganga icyo ukora iki, you scratch just guhunguraho selile nkeya ukazoshyira kuri slide, microscopic slide, then ukohereza muri pathology bakareba are these cells metaplastic or just or dysplastic or already there is a cancer. So niyo mpamvu nzamubona bandika bohereza umurwayi akaba bati umurwayi tuwohereje twakoze colposcopy dusanga afite aceto white. Aceto nuko buresha bakoresha acetic acid, ni kimwe muri ibyo bakoresha bareba ko haba changes barebesha amaso. Iyo bavuga aceto white ni ukuvuga ko ntihagombye kuba white, hagombye kuba brown. Ibaye white ni ikibazo. We saw aceto white lesion, so therefore we took cells, we want you to see if there is a cancer or precancer lesion. So ni yo basis yabyo. So murumva metaplasia y'ubwayo iba yahinduye character ya nature of this cell, bifite implication biochemically, because the cells, if these are rich in glycogen, these will not be rich in glycogen. Therefore, they will not be brown when you apply acetic acid cyangwa se izindi solutions zirimo iodine. And when the dysplasia is the same, ariko dysplasia ikindi ushaka kubona kuri colposcopy, you see many blood vessels, uzibonamo munsi ko ntabwo uzamure epithelium, ariko ubuzibona ko zirimo hasi kubera urumuri rwinshi bazibona. So at this dysplasia then, birumvikana umurwayi umudamu uzaza afite dysplasia ntabwo rendezvous zamuha ari kimwe n'ufite metaplasia muri no. So there are protocols. Nowadays we use a protocol yitwa Bethesda. Bethesda ni umujyi wo muri United States, habereye conference. Kenshi protocol dufite zitirirwa umujyi zemerejwemo. Habereye conference baremeza bati okay, umudamu uzaza akaba mukabona ni normal na kuri colposcopy na ya bashyizeho na ya acetic acid cyangwa se iodine solution na cytology kaba normal. So ubu ngubu ashobora kugaruka nyuma y'imyaka 3. But ubu hari ikindi cyongewe ndakivuga nyuma gato. So bakavuga nabwo usanga ari metaplasia wenda azaza nyuma y'amezi 12. Nimusanga ari dysplasia, either niba ikiri hafi ko ziri mu bwoko nazo, iyo izi changes ziri muri 1/3, lower 1/3, icyo gihe tuyita ko ari grade 1. Iyo zigeze muri 2/3, grade 2, yaba ari ifata wall yose ya epithelium bita grade 3. So bitewe naho ageze bamubwira either niba ari muri grade 1 ukamubwira y'amezi 3, niba ari grade 2 wenda nyuma y'amezi 6, niba so niba ari after grade 2 yagapfa nyuma y'amezi 3, niba ari grade 1 akagapfa amezi 6, niba ari grade 3 ugomba kugira icyo ukora ako kanya, either gukuraho ako gace kose gafite ikibazo, kuba wasizeho acid ubona ahabaye aceto white, aho region iri ushobora kugakora ibyo bita conization n'urugero. Usuba ako kanya uramuprograma muri icyo cyumweru cyangwa mu byumweru bibiri ukaba wabikoze. So this is how things are done. So I was saying that hari ikindi cyongera mu iki gihe nuko ukora na test ya HPV, human papilloma virus. So when you see that that woman is infected by HPV, then hari ibindi ugomba gukora. So protocol icyo yacyongeyemo ubu ngubu nacyo. But the basic is that Bethesda system. So we arrive to dysplasia. So dysplasia, once it is there and you don't act, nta kindi kizakurikiraho, risk hari nuko azagira cancer. Gusa ntabwo abantu bose bafite dysplasia ba progress, ariko risk iba yiyongereye cyane ko azagira cancer uyu nguyu. So that's why you have to act. Ntabwo nk'umuganga ushobora you cannot predict, say, so if you have 10 women with dysplasia, uvuge ngo uyu nguyu ndabizi ashobora kuzagira cancer ngo ariko ntihajya. No, you can't predict. So there are many studies undergoing, bareba izindi factors bakareba ko bashobora guprogressa n'abatabishobora, ariko that's at the epidemiological level. So muri public health kuvuga ngo indwara yafashe umuntu umwe mu bantu 10,000 niba atari indwara yandura cyangwa iki, public health ntabwo ari ikibazo. But when you are in clinical medicine, in medicine itself, it is a big issue. Nta murwayi n'umwe wo kunegereje, nta kintu na kimwe cyo kunegereje. But at public health level, kuvuga ngo hari virusi yaje yanduza ariko ishobora guhitana wenda umuntu umwe muri miliyoni, ntibisanzwe. Na gripe mu Burayi ihitana abantu batari bake, hano ishobora no kwica nta n'ubyita kuko bizwi atari bizikomeye, nta n'ubitinda. This is at public health level. But you can have a small child suffering with that flu. You must act. You must act because as a medical doctor, your scope of the career is that one person. It is not 12 or 13 millions of Rwandans. Umurwayi umwe niwe ushinzwe ako kanya, ntabwo ureba at public health level. So then after dysplasia, now we see there is invasion. Invasion, I had explained this. The basement membrane here is breached by some cells. So when these cells go down, usually they will go into blood vessels or lymphatics and so on. Iyo zinjiyemo rero zishobora kwinjira mu maraso cyangwa muri lymphatics zikajyana nabyo aho lymph ijya cyangwa se aho amaraso ajya. Ubwo usanga umuntu avuga ngo afite kanseri y'igifu ngo ariko ngo yamaze kugaba amashami mu mutwe, mu mwijima, mu bihaha and so on. Naho ngaho biba byaturutse. I hope I'm explaining where you can understand. So you will find that there are somebody who said in cancers, cells have forgotten how to be good neighbors. So they are not good neighbors. That's why they destroy whatever they meet. Nta kintu na kimwe ari bones, amagufa rero tuvuga comment, they destroy. The tendons, they destroy everything they can destroy. Cells have forgotten their identities and functions. I think you can understand it from even in metaplasia and dysplasia, because these cells are usually, as pathologists, even the most experienced professors of pathology, this is a cancer, yes, ukabibona ko ari but ukaberwa zakomotse kwihe seri, kandi mu gihe cyose utamenya seri zakomotseho ntabwo ushobora kumwohereza kuri oncologist. Ntabwo oncologist ashobora kuvura kanseri atazi serire yaturutse, ibyo ntabwo bishoboka. Kumubwira ngo genda bibona neza ari kanseri, ibyo ntacyo bimubwiye. You must say, is this cancer arising from epithelial cells? Is it from lymphocyte? Is it from skeletal muscle? Because treatment options are quite different. Ntaho ziba zihuriye. And if it is a epithelial cell, ntabwo uzamubwira ngo okay, we have seen a metastatic, a cancer having yagabye amashami niyo twita metastasis, yageze mu bwonko cyangwa mu mwijima, kuvuga ngo I see it is a carcinoma. No, carcinoma azaba ati okay, carcinoma ni nyinshi. Ese ni yavuye muri prostate, yavuye muri breast, yavuye muri stomach, yavuye muri colon, yavuye muri ovary, muri cervix and so on. Ibyo byose azabibaza kubera ko nubwo zose ari carcinoma, ariko buri yose iya prostate ntabwo ivurwa nk'iya breast cyangwa se ngo ivurwe nk'iyigifu. So I want to know the origin, the primary tumor. So you will see usually in our report we say, okay, we find this is a tumor, but we have to work with clinicians and the radiologists to know the primary. As far as the primary tumor is not known, no treatment to that patient. So it happens this last Friday, we had a visiting a professor from California. He was teaching a postgraduate program, and then so he told us a history which happened to him when he was just a young pathologist, no, a resident, a postgraduate student. So he received a patient, then examined the stomach. So they found a tumor, they did biopsy. So he reported this is a carcinoma, a cancer of the stomach. So they did the surgery, and the patient was very fine for 5 years. After 5 years, then they found in the axilla, they found a nodule. They took biopsy. So the histology was exactly as what they had described before in the stomach. I just say, but the cancer of the stomach does never send its cells to the axilla. So what happened? So somebody started to say, probably this was a breast cancer, not a stomach cancer. So ariko uwo murwayi nta region yagaragaza muri breast. So when they went for mammography and CT scan, they found there was a tumor, a very small tumor in the breast. And when they moved it, it has the same features as the one in the axilla, lymph node, and as in the stomach. So they went on to do molecular testing. They found that the cancer which was in the stomach was not originating from there, was actually from the breast. But he was telling us the patient didn't cause any problem. She was happy. She did not complain because they explained everything, so she understood. But there are some who are troublesome, saying, so why, as medical doctor, you failed to know this was coming from breast? So this is to say that to know the primary region is very important. Kubera ko umurwayi n'ubundi bari bamuha imiti yo cancer of the stomach, uretse kumuhaga n'ubundi bamuha imiti ntacyo yakoze, ahubwo muri breast kakomeje gukura gatuma bijya muri axilla. Iyo baza kumenya ko ari breast hakiri kare bakayivura nka breast, ntabwo ari kugera aho hose. So this shows the importance of knowing the real primary tumor, where the cancer started. So the cells have forgotten their identities and their functions. Ngira ngo tuzabigarukaho cyane cyane muri systemic pathology, aho usanga umuntu afite cancer of the, let's say, cancer of the lungs, ariko akagira hormone nyinshi nk'izikorwa na adrenal gland, because the cancer cells in that lung cancer have now acquired the identity of adrenal gland cells. Ikabyiha igakora zira hormone corticoids zikorwa na adrenal glands. Ku buryo when you receive that patient ugasanga afite Cushing syndrome, wenda za aldosterone nyinshi n'ibiki byose, you will automatically think, okay, probably this patient is having a tumor of the adrenal gland. You do CT scan, you do MRI, you find nothing. So when you are then a good medical doctor, you will think about that aspect, that probably it is a cancer arising from another area which has changed its identity and is behaving like as if it is coming from adrenal gland. Being able to be paraneoplastic syndrome. So here we have now pictures, examples. So this is written in French. So example of neoplastic growth. So goiter, so what is the cause? It is caused by iodine deficiency. When this patient gets therapeutic iodine, this goiter will disappear. Of course, when the goiter has undergone many changes, including calcification, necrosis, hemorrhage, ntabwo ishobora kugabanuka, birumvikana harimo fibrosis, so icyo gihe barayibaga. But when there is no fibrosis, usually they give this patient iodine. So when the iodine is there, then that goiter will regress, because this was not a neoplastic growth. It is non-neoplastic growth. So once you remove the cause, the tissue will become normal. So this is on the left, we have a normal thyroid. You have the right lobe, the left lobe, and the isthmus in between. So you see 4 centimeters where they are. But when you come here, you see that this is very big. And in our country, we have many patients with goiter. So at least each day in the pathology, we receive those patients with goiters or tumors of the thyroid. So there are many. There are risk factors which are debated, of course, for this. Kubura iodine ni kimwe, ariko ni iki gituma umuntu abura iodine? cyane cyane ko umunyu ukoreshwa mu gikoni baba barongeyemo iodine. Nicyo gituma umuntu abura iodine muri. So there are things maybe you have learned even in at high school, cyangwa muri chemistry muri na first year at university, that usually the problem here, deficiency iodine, is not because of diet. It is the issue. Okay, suko iodine yabuze muri diet, ahubwo nuko muri diet mu byo abantu barya hashobora kuba ibindi bintu birwanya iodine cyangwa iode. So biravugwa ko harimo ions bita cyanide cyangwa thiocyanide. Izo ions iyo ufashe ibiribwa nk'ibinyobwa irimo, when there is iyo bigeze kuri absorption in the intestines, habaho competition. Muzi ko buriya harimo za receptors ibyinjira n'ibisigara, so habamo competition. Iyo rero bihuye burya iodine iratsindwa, izo zindi akaba arizo ziba absorbed mu cyimbo cya iodine, ko bifite ukuntu bijya gusa. So iodine rero igatakara muri stools, ibiba absorbed, bikaba n'umubiri udakeneye, ahubwo biteza ibibazo. So ni uko nk'uko bivuga. So there are some foods which are known to have a thiocyanide and cyanide. So usually niyo baje barwaye umubajije akenshi ukamubaza urya ibi n'ibi aravuga ati narabiriye cyane, ndabiriye cyane. So ugahita ubona ko it is true. So another example of non-neoplastic growth is hypertrophy that you have seen in the first chapters. So this is a ventricle of the left ventricle of the heart, and here you see that lumen zabaye ntoya, ventricle ari okay, left na right ventricle zabaye ntoya, ahubwo thickness iriyongera cyane. So this is hypertrophy. But if you remove the cause, for example, abashobora kubigira ko yabitewe n'uko afite hypertension, n'umuvura hypertension, iyi hypertrophy izahita ivaho. Muzaba mubibona, it is just simple. Munaza muri district hospital ndafite hypertension, ibibazo by'umutima, umukorera chest x-ray, uzabona ko umutima wawe munini cyane kuri igihaha cy'ibumoso akenshi ubona cyabaye gito cyane, ndetse ahagana hasi ukaba utakibona, umutima warahiganje. So this is because there is hypertrophy for this muscle. So when you treat, you start now treating hypertension, akagira normal blood pressure nubwo akiri ku miti, the hypertrophy will regress. Uburyo nukora x-ray, control x-ray, uzasanga umutima wawe muto. So that means also it is non-neoplastic, because if you remove the causing factor, the change will also regress. So it is a good.
So lecturer.
Yes.
Ntabwo numvise impamvu nyine iyo umuntu ashobora kugira hypertension nyine mu mutima ukabyimba.
Okay, ni kimwe nuko ukora physical exercises, nukora physical exercises uterura ibintu, muscles zawe zirabyimba. Umuntu afite hypertension, umutima ukoresha imbaraga nyinshi ngo wohereze. Niba ukoresha imbaraga, its muscles must increase in size kugira ngo ushobore kohereza amaraso cyane ukoresheje za mbaraga usa. Hypertension means there is high, hari imbaraga nyinshi, umutima ukoresha imbaraga nyinshi, nicyo bisobanura nta kindi. Mu kuzikoresha rero muscles zizaba hypertrophied. It's just simple, nkuko uterura ibintu the cells zawe ziba nini. It is just a mechanism, a defense mechanism or adaptation.
Ndabyumvise.
Okay. So this is another example, but this is a tumor ovary. So this is the size of the ovary. It is very small. You see, it is about 4 centimeters in length. But you see this one, 4 centimeters causing an itch. So, and I would say that the ovarian tumors, they are the largest tumors we see even in our country. I think there are the specimens we brought from Sashka to the lab of the school at the campus. So you will see some of these. Nyango tuzabireba byinshi cyane muri systemic pathology, organ per organ. Ariko muzasanga harimo binini bari mu ndobo nini, hari usanga ibiro 13 and so on. The normal ovary is just a few grams. So this is a tumor. Of course, it cannot regress because it is a true neoplastic condition. So this is a uterus. So umuntu wenda yayikase kugira ngo urebe muri lumen yayo imbere. So in its lumen, so here we have, this is the cervix and the cervical canal here. And then this is the endometrial cavity. Ariko endometrial cavity ibi isa na brown ubundi, kandi iri smooth. But here you see things which are looking like fingers. Ibi byatewe n'iki? Nuko endometrial yagize hyperplasia. Usually we don't use the word hypertrophy because when you go to cytological, hyperplasia ni ijambo ntabwo wayiconfirminga kuri n'amaso, it is histology. So you see that the cells have increased in number, the glands have increased in number. So because of that, ikagira bimeze nka polyps, finger-like structures. So this is also seen in many cases, endometrial hyperplasia, and the lady will come to you saying, I have a bleeding all over the time. So even outside the menses, I continue to bleed. So because of this hyperplasia. So endometrial hyperplasia is the most common reason of having this bleeding in women, but there are other causes. So there's a benign prostatic hyperplasia. So by benign, understand it is not a cancer.
Excuse me, lecturer.
Okay.
Excuse me. The one we saw in the ovary and that endometrial hyperplasia are benign.
They are benign, of course.
Yes, thank you.
Okay, I didn't explain much here. This we shall come back on this. Cystadenoma, because by its name here, it means benign. Tabigarukaho nyuma, ntabwo ibintu byose twabipresent icyarimwe. So biragaruka inyuma uburyo tubiha amazina, kandi izina iyo uyihaye izina bihita byikumenyesha ni benign or malignant. So endometrial hyperplasia is benign, but it is usually when it persists, cyangwa uje ku muntu uri gukura ukuze imyaka 60, when nothing is done, usually these women, they have risk to have endometrial cancer now. And on the other way, harabagira atrophy. Ugasanga endometrial isa naho ntayafite. These also, they have the risk of developing another type of cancer. So there are two types of carcinoma anyway, endometrial. So this is benign in men, of course, because they are the ones having a prostate, kandi nyango ni ikintu cyiganje. Kugira ngo kuva ku myaka 40 urebye abagabo benshi baba bafite hyperplasia. Bamwe bagira risk yuko bashobora no kugira cancer guhera ku myaka 55 ku buryo ku myaka 70 ubundi avuga ko abantu hafi 70%, abagabo 70% by'abagabo bafite imyaka nibura 70 buri aba bafite cancer ya prostate. Tuzabigarukaho, ariko iyi cancer ya prostate ibyayo nyine birimo ibintu byinshi, harimo ibintu factors nyinshi zituma kugira icyo igikorwa bigorana. Icyambere ni ukubera ko abenshi bagira izo cancers bakuze ziba ari low grade, ntabwo zikura ngo vuba ngo zishwanyaguze, bituma rero rimwe na rimwe iyo uyibonye uravuga uyu murwayi n'undi abenshi birazwi muri literature ko ashobora kuzapfa yishwe n'ikindi kintu atari yo cancer. Kujya kumubaga ko ahubwo beramwongera bazo kandi wenda cancer nta n'icyayi kuzamutwa. So aho hazamo dilemma. So I remember when we were developing the protocol for managing and screening prostate cancers in Rwanda, when we tugeze kuri prostate cancer haje discussions, and icyumweru cyose izindi turabikora, ariko prostate birananirana kubera ko ntabwo twashoboye kumvikana ku cyakora. Kuko kuvuga ko umuntu wese ubonye prostate cancer agomba kubagwa, no mu bindi bihugu ntibabikora, n'ahandi hose biracyari ikibazo kumvikana kuri protocol y'igikora. Gusa ubu hari ibyumvikanwaho, niba history yavuze ko ari high grade, meaning it is aggressive, uwo nguwo you must act. Cyangwa se niba ari mutoya, akiri muto, ariko niba akuze ni low grade, kumubaga ushaka kumuza complications akabaho nabi, wenda afite ibibazo incontinence urinaire n'ibindi, kandi utamubaze nta nicyo byenda kumutwara. So there are many challenges here. So as you come aboard among the healthcare professionals also, you will contribute maybe to that. But hyperplasia, ikibazo cyayo rero, iyi ngiyi ishobora kwica kubera iki? Prostate uko iteye murabizi ikikije urethra. So gukikiza urethra bivuga iki? Benign prostatic hyperplasia is not a cancer, is benign. Ariko itangirira muri center prostate. Center ni naho hari urinary hari ureter. Iyo rero haje benign prostatic hyperplasia igakura, it compresses the urethra. The urethra being compressed, the urine will not pass. So the patient will have a urinary retention. Niba nta gikozwe, uko umubiri uteye nuko urine, when it is there for time, there are many risks. Hari kuba yateza infection, ariko risk nini nuko yuzura zigasubira hejuru zikajya muri kidney, zikangiza kidney, umuntu abangabo basanga yishwe cyane cyane na renal failure kubera icyo gusa. So otherwise iyo bimenyekana ko ayifite, uzasanga ari umuntu ufite sonde cyangwa catheter banyuje muri urethra ahorana kugira ngo urine zisohoke. Nka bya bindi bya coronary twabonye bya dilatation boherezamo balloon n'ibikini kimwe n'ahanga. So akayihorana bakayisimbuza kugira ngo atagira infection, wenda mu byumweru bibiri bitewe na protocole bakurikiza. Hari n'ubwo usanga harifunze burundu idashobora no kwinjiramo, ahubwo bakayinyuza hejuru ya pubis, suprapubic catheter. Ibyo bibaho. So birumvikana benign prostatic hyperplasia is a benign, it could not kill, but because it compresses urethra with the consequences, then is very easy. It can kill someone indirectly. So and another thing I would say here to when that's muri systemic pathology, nuko cancer yo ntabwo itangira muri center, no, it starts on the periphery. So akenshi umurwayi ufite ntabwo ibibazo bya urinary retention cyangwa incontinence and so on, keretse yabaye advanced ikinjiramo cyane kahashwanyo. Otherwise ahubwo bagutanga kuri periphery, yukoze touche rectal digital examination, wumva ko prostate yakuze, iri rough, itari smooth, iri hard. So ahango niho bahera bagakora biopsy to confirm if there is a cancer or not. And another problem is that because it starts at the periphery, usually it can destroy the surrounding tissues, including the rectum, which is just beneath it. So this is benign prostatic hyperplasia. When you cut, you see that the tissue is homogeneous. So as I was saying, but this is a true picture. The benign tumor here, you see that it is very delimited. So this tissue here around ntabwo ari part of the tumor, because the surgery buri gihe ushyiraho marriage. Tumor ntabwo ivanamo yonyine, uvanamo na a part of a normal tissue to ensure the tumor is completely removed. So iyo udashyize izi marriage cyane cyane atari cancer, nibwo mubona abaganga bajyanwa mu nkiko. There are just simple things like this. Mwamwarizo mwishwenda mu cabinet so tukuba ko umuntu wese adashobora gukora amakosa, ariko hari amakosa atagombye. Hari insida zibaho, tewe umuntu anesthesi agize reaction arapfuye, ntabwo ari ikibazo cy'ikosa cyangwa cy'uguswa. It is an incident which can happen to anybody. Ariko niba ugiye kubaga umuntu cancer ukayinyuramo hagati ugasigamo igice, this is a mistake. So that's why here there are normal tissues. So here we have a capsule, a white tissue one, delimiting the tumor itself. So you see that the tumor is well circumscribed. It is of smooth surface. So how about cancers? This is a cancer in the lungs. The normal lung tissue is here. Ubona ko utuntu kubera ko harimo alveolus. Ubona ko atari solid tissue as per se. Ubonamo ko ari nk'ikintu kimeze nka matora and so on. But here we have a solid tissue, whitish. So this is a cancer. It is not very limited. You see that the surface is not regular. It is irregular. It infiltrates, and probably there are here areas of hemorrhage, or the hemorrhage, black, and the ongoing hemorrhage. So multiple adenomas of the colon. So muri colon ashobora kujyamo adenoma. Adenoma itself it means benign. Nkuko nabivuze turabigarukaho nyuma. So usually there are multiple lesions. Usanga they are well circumscribed. They don't cause any other problems. Usibye ko zishobora gutuma stools zidahita kubera ko zishobora kufunga with the consequences. Otherwise, no, they are not cancers. But there is a syndrome called familial adenosis poly, FAP. Naho usanga umuntu afite adenoma, utu tubyimba twinshi, utu dupolyps twinshi mu mara ukoze endoscopy. So iyo bifite rero tuvuga ko afite familial adenosis poly, nuko afite mutations in their family. Hari mutation bagiye bahabwa n'ababyeyi babo. So in that case, then when you see that nubwo atari cancer, but when you don't act, this patient will develop a cancer in the future. So ntuzasanga bavuga ngo uyu muntu twabonye afite adenosis poly, therefore we have done colectomy, bagakuramo igice cya colon zirimo izo ngiro. So ngira ngo cut point cut off bashyizeho nuko bibonye polype zishobora kuba zigeze ku 100, you must remove the colon. If they are less, usually ni ikintu kitari familial, so bishobora kuba ari ibintu sporadic bitazanamutera cancer, but you must of course make a follow-up of such cases. So here it was just to compare this benign and this malignant tumor. So this is the uterus. They used to have the large tumor, the well circumscribed tumors. These ones, these are nodules. Akenshi usanga nafitemo bwinshi cyane. So these are benign tumors arising from smooth muscle. So we call them myoma cyangwa fibroids. Ni terime y'abaganga in general fibroids, but the more technical term, especially for pathology, is leiomyoma. We shall see why it is called like that. So turabibona ryo byinshi cyane, especially biza kuba this happens to ladies who got their pregnancy at a very advanced age, or who have not acquired a child. Abatarabyaye cyangwa se batinze kubyara nibo bakunze kubigira, ariko n'abandi bose bishobora kubaho, ariko abo nibo bagira risk nini. So umvikana if you receive a woman nk'umubikira ni urugero, cyangwa umukobwa atinze gushaka cyangwa washatse ntabyare ariko akaza afite a bleeding, genital bleeding. So of course you will examine the cervix. If the cervix is normal, you do an ultrasound echography, you see if there is endometrial aplasia, and at the same time you see the myometrium if there are nodules. So either there will be endometrial aplasia or there will be these nodules. These are the main reasons for bleeding from the uterine body, not the cervix itself. Of course, another cause is cancer. So this is a cancer of the lip. You see how it has destroyed this part of the lip. It is black in some areas because of hemorrhage and
Lecture question.
Yes.
There is hari ikibazo twariho twibaza kubera iki bibaho nyine ku bantu batabyaye gusa. Kuki aribo badevelopa doze kayido?
Ntabwo navuze ko ari gusa nibo bafite risk nini, navuze ko n'abandi barabigira ariko bagira risk nini. Okay, there are things you can explain and there are others you cannot explain. Ariko icyo uhita wumvamo this is a matter of hormones. So aha bivuga ko umuntu wagize exposure kuri progesterone, risk zo kubigira ziragabanuka. When you are someone is pregnant or she's breastfeeding, your progesterone is high in her blood. So progesterone will be, it is protective against many things, including breast cancer. So uzasanga abantu bagira breast cancer akenshi ariko si uko ari bo bonyine, abantu batinze kubyara cyangwa batinze kubyara cyangwa babyaye ntibonse, so risk ziriyongera because estrogen is the risk factor to these things. And when you have a progesterone, it protects you against them. There is no other way to have progesterone except to have a pregnancy and breastfeed. Icyo gihe nibwo progesterone ijya hejuru. So this is how it is explained. So the cancer, it destroys. So on histology, this is still the papilloma. It's a benign tumor, maybe it is histology. We don't take much time. Ariko hanga cell ubona ko zabaye mbi. This one cell, this is another cell. Twebwe hari ibyo tureba. You know, mitosis. Buri gihe turanazibara muri tumors, especially when there are cancers, we must count them. Dufite protocol tugenderaho tukaboti wenda kuri cancers arising from non-epithelial tissues, you must count maybe 10 high power fields of the microscope. Nubonamo mitose wenda 10 bizaba ko ari high grade. Niziba munsi y'ebyiri, this is most likely benign and so on. But there are other things we see. Mitosis uko mwayize igira it is bipolar kubera ko cell zagabanywamo kabiri. But in cancers, it is tripolar. This has this pole, this, and this. So this, when you see tripolar mitosis, we call it atypical mitosis. It is a sign that it is no longer a benign condition, a normal condition, which is a cancer. So, ngira ngo tugiye kuri iyi part ya nomenclature, uko tubiha amazina ari byo nyine ntagiye ntindaho nabivuze adenoma, cystadenoma ni ibiki, so nuko narinzi ko tubigaruka hano. So any questions so far before we proceed?
No question.
No question, okay. So I know globally this domain, the domain of cancer is ubu ngubu abantu bakishyiramo focus si mu Rwanda gusa n'ahandi, cyane cyane ibihugu byateye imbere aho baba bafite amafaranga menshi yo kwa research. So ni cancer ni ikintu gisa naho kiri gushyirwamo ingufu muri iki gihe. So mu gihugu cyacu kubera resource limitations, focus zikorwa kuri cancer zimwe na zimwe, na breast, muzamumva breast na cervix cyane cyane, n'ibindi bijyamo, ariko izo nizo cyane cyane. Ariko icyo nshaka kuvuga nuko hari opportunities nyinshi mu gukora research, kubiga komeza kwiga kujya muri izi fields. So ari ukujya muri basic sciences related to cancers, ari ukujya muri bijyanye na pathology, diagnosing them, categorizing them, to predict which one can be curable or not using molecular or cellular aspects. Hari n'abajya muri part of oncology. Oncologists, they just treat cancers. Hari n'abajya muri surveillance yazo, public health aspect of cancer. So all these fields. So hari n'ibindi byaje bungu bya genetics and bioinformatics. Na mu Rwanda ushobora kuba dufite nka bioinformatician umwe cyangwa babiri and so on, but cyane ko ari field tutaratangira gukoresha ni humans as it is done in other countries. So I want just to say that there are many opportunities about research on cancer, so grants from different corners and so on. So those interested, they can just be they can start thinking about this. So nomenclature, as you know, nomenclature you had, this is what you had in chemistry, in undergraduate, in secondary school, high school. Nomenclature, uburyo mwita ibintu byo muri chimie and so on. So hano rero mu buryo twita tumors. How do we give names to tumors? And when you give a name, it has implications. So how do we give names to tumors, not only cancers? So the first thing is to know the cell of origin. Then to that cell of origin, you add a suffix. So cell of origin, it depends. If it is a fibroblast, if a tumor is arising from a fibroblast, so harazamo ijambo fibro, because it is a fibroblast, then wongeraho suffix. So suffix uzongeraho izaterwa niba is that tumor benign. If it is a benign, you add oma. If it is malignant, you add sarcoma. So therefore, a benign tumor arising from fibroblast will be called fibrooma. But the one O is deleted or omitted, so you just say fibroma. If it is a cancer, you call it fibrosarcoma. So the suffix sarcoma is used only for cancers arising from mesenchymal tissues, not epithelial cells. And for epithelial cells, we use the suffix carcinoma. So you have seen where it is an adenocarcinoma. Carcinoma, means it is a cancer arising from epithelial cell. Then, depending on the tissue of origin, usually what? Is it adenocarcinoma from stomach or from colon? If you when it will give a diagnosis. But here, histogenetically, we give the names to these tumors. So the suffix areoma for benign tumors, carcinoma for cancers from epithelial cells, and sarcoma for tumors arising from cells which are not epithelial cells. Of course, we have some exceptions. I will present them thereafter. So fibroma, it is benign from fibroblast. Fibrosarcoma, it is cancer arising from fibroblast. Os, you know, os in French, of course, and Latin and so on. Os means bone. So when it is a tumor, a benign tumor arising from a bone tissue, osteoclast or osteoblast, and the osteocyte, it will be called osteoma. Ost for bone and oma, benign tumor. If it is a cancer, it is osteosarcoma. And then we have now tumors arising from epithelial cells. Musical epithelial cells, some they are just lining, covering epithelium. Hakaba na epithelial cells which line glands. So with that, so line glands or secrete. Nkaza twabonye muri cervix and cervix, they are secreting mucin or mucus. Ariko squamous cells out, they don't secrete mucin. So those which produce mucin, then usually any tumor is komoka ukoresha ijambo adeno. So any cells which have a character of glands, gland murazi is there can be exocrine glands, like pancreas, salivary gland, izo muri stomach, izo mu kanwa, and so on. So these are exocrine, and the endocrine, of course, zivamo or more. So buri yukoresha ijambo adeno. So adeno refers to gland, because the gland is always a tumor arising from gland is almost always of epithelial origin, then has a ijambo carcinoma when it is a cancer. So adenocarcinoma is a cancer arising from epithelial cells of glandular aspect or glandular type. So the word adeno. So when it is a benign tumor arising from a gland, you say adenoma. So therefore, can I say adenoma of the salivary gland or adenocarcinoma from stomach or salivary gland and so on. So this is when the cell of origin is glandular. When it is not glandular, ho hazamo utuntu dusa n'uturuhije nubwo ari byo byiganje. So papilloma biterwa nuko igaragara n'amaso. Ayo yitwa papilloma imeze nka choufleur cyangwa se finger-like structures, cyangwa imeze nk'utubabi and so on. So icyo gihe iba ari papilloma. But this one, it arises from squamous cells. And when it is a cancer, we call it squamous cell carcinoma. So yeah, rya jambo squamous cell ryo rigarukamo nta yindi shusho ihari aha ngaha. There's no other choice. We use the whole one. Squamous cell carcinoma of the cervix, squamous cell carcinoma of the penis, squamous cell carcinoma of the skin, and so on. Of the lungs. So if it is a tissue of cartilage origin, benign, chondro, it refers to cartilage. So it will be chondroma if it is benign, and chondrosarcoma when it is a cancer. Sarcoma because it is not a epithelial cell. It are mesenchymal cells. So benign tumors, ngira ngo ahangaha ni these are like exceptions. We have, when we started, I said the benign and the cancer and the malignant tumors, they are monoclonal. Ariko navuze ko ari exceptions. So hari tumors zifata ziza muri breast. Zikunda kuboneka ku bakobwa batoya imyaka 20, ntekereza ko nk'abari muri karasi aho ngaho, muri iyo age yabo bagomba kuba careful. So bita fibroadenoma. If you understood well what I explained in the previous slide, there is the word fibro, referring to fibroblast, and there is the adeno, referring to gland, and the oma, benign. So it means this tumor combines both fibroblast and glandular epithelial cells. So you don't have ari monoclonal, the exception, it is polyclonal. It is a tumor involving both the fibroblast and the glandular epithelial cell. So it's called fibroadenoma. So it is very frequent in girls around 20 years. Tutubyimba rimwe na rimwe byinshi cyangwa se binini, can be unilateral or bilateral, but these are very frequent. Abenshi baza kubera awareness wenda batangiye kugira ubwangu buri kanseri, ariko abandi bakaza kubera ko bibatera aesthetically ntabwo biba bigaragara neza, ribyimbye cyane ku buryo ushobora no kugaragara anambaye imyenda ko hari ikibazo gihari. But it is good always when someone has such changes to go to clinics, to hospitals, bakamurebera ikibazo gihari. So baraza hafi buri munsi tatu bafite, and the first thing we do final needle aspiration. We insert needles and we take some cells, we see if they are benign or not. When they are benign, then we say this is probably fibroadenoma. So the breast surgeon will now decide if they do surgery or if they just wait and see what will happen afterward. So the polyps of the colon also, it is the same. The polyps, they involve both mesenchymal cells and the epithelial cells. So they are like a polyclonal. So then, hari andi mazina tutarebye, lipoma, lipolipids. So this is a benign tumor arising from adipocyte or cells producing lipids. So this is the lipoma. When it is malignant, we call it liposarcoma. So ngira ngo ahangaha ntabwo tuzatwumva umuntu azavuga ngo uyu murwayi ngo afite adenosarcoma. Adenosarcoma doesn't exist. Adeno, it means a gland. Sarcoma, it means a benign tumor arising from mesenchymal cell. So a mesenchymal cell cannot produce a gland. So icyo gihe hazaba hari ikibazo. Hari exceptions tuzi wenda ntari gutinda ahangaha ushobora gusanga umuntu ako they can combine both carcinosarcoma, it exists. Ariko icyo ukayita gutya, ariko nabo bamwe bafite ngo adenosarcoma. Njya babona and very special experienced specialists in surgery and so on bakavuga icyo jambo nk'iki. So notekereza ubivugiye mu nama mpuzamahanga harimo abantu bavuga ati this is a person is representing Rwanda here, bakavuga icyo jambo nk'iki. Nk'utagiye mu ishuri. So ibi ni ibintu cyambere biroroshye kubyumva, kubizaho umutima no kubyapurika muri niba routine activities, clinical activities. Ntabwo ari ikintu gikomeye, ntabwo ari molecular, ni kwita izina ukamenya ngo iki kintu ubundi gishobora kwitwa gutya cyangwa se nkoresha irihe zina. And when you are not sure, then ask your colleague pathologist or even students in pathology, the postgraduate. So instead of using terms which will make you that more clear. So as I was saying, carcinomas, they are malignant tumors of epithelial cells. So hari ibindi turi bugarukeho cyangwa wenda tutanatindaho, hari amagambo dukunda kongeraho when we give a diagnosis. We say well differentiated, moderately differentiated, and poorly differentiated. So when we say well differentiated, ni ukuvuga ko usibye ko amasere nyine yateye kanseri ariko asa neza na the cells of the corresponding normal tissue. So niba ari squamous cells well differentiated, squamous cell carcinoma well differentiated, uzasanga amasera asa na skin. Bitandukanira hehe ko they destroy basement membrane, they entered into blood vessels of lymphatics, and they went maybe far in the other organs. Otherwise, ukabonye karaho gusa batakubwiye history, wabona this is just a normal epidermal cell. Moderately differentiated, some of the cells will have features of a mature tissue, normal tissue, but others will not. While for poorly differentiated, it will be very difficult for even a pathologist to know, is it this cell from stomach, is it from the liver, and so on. Ahangaha rero tuzakoresha advanced methods, immunohistochemistry, spatial stain, molecular studies to know if these cells came from breast or not, and so on. So have a squamous cell carcinoma, adenocarcinoma, adeno from glands. So had it been it can be alveolar, n'ibikora ibintu bimeze nka alveoles of the lungs. It can be papillary, bya bimeze nka chou-fleur, cauliflower. We call it cauliflower in English. Tubular if it forms tubules. So anaplastic and undifferentiated, usually it falls into this category of poorly differentiated. So then there are in other areas aho burije navuzo kare burije dushiraho organ yaryo. If it is a carcinoma arising from the epithelial cells of the liver, burije tuvuga ko yitwa hepatocellular carcinoma. So hepatocellular, because here we have hepatocytes in the history. Then a cancer arising from them will be hepatocellular carcinoma. Ahangaha ubu twitandukanye na cholangiocarcinoma, iyi iva muri za bile ducts. Bile ducts maybe it can be even intrahepatic. And this is important for us and the oncologists. Bakuvuze ngo nabonye carcinoma of the liver, nta kintu kimwe uzaba uvuze. Niba udashoboye gutandukanya ko ari hepatocellular na cholangiocarcinoma, nta kintu uzaba ukoze kubera ko hepatocellular carcinoma, until now, we can say it is not treated. Ariko cholangiocarcinoma barayivura, umuntu ugera akaba yakira. So nuko abanye ntanze diagnosis ko umuntu afite carcinoma of the liver. Nta kintu uzaba ukoze. It's just confusion. I must precise if it is cholangio, therefore the patient can be treated, or it is a hepatocellular carcinoma whereby the patient was canceringa, then atahe cyangwa se rimwe na rimwe bagwe no mu bitaro. And even last week we had the case of hepatocellular carcinoma, umuntu waje ku bitaro sashika, so yaje iri advanced, yagize hepatite C virus n'ibindi bishobora kuba ari yayimuteye ko mu yarakara ko ayifite iri active mu maraso. So she just stayed in the hospital not 2 weeks, less than 2 weeks. And then she died last Monday. So bari bakibona iyo bigaragara ko ari cholangiocarcinoma, something could have been done, but for hepatocellular carcinoma, when you diagnose it, ntabwo akenshi umurwayi muri amezi menshi. So I am just explaining these things, but also giving some implications on why these names matter and categorizing them, why it matters a lot for the patients, for the clinicians, meaning the medical team or healthcare team who is caring for that specific patient. So for sarcoma, as we have seen, chondro for cartilage, chondrosarcoma, osteosarcoma for bone, hemangiosarcoma from blood vessels. Glioma, these are exceptions. Tumors arising from astrocyte bazita gliomas in general. They have many subtypes. So akenshi muri rusange tuzita astrocytoma, but when it is malignant bayita glioblastoma. So hano rero hahise indi suffix tutakoresheje mbere, blastoma. So whenever you will find a tumor bakongera ijambo blastoma inyuma as a suffix, ni ukuvuga ko it is a tumor igaragara nkaho ari embryonal tissue of that organ. So uvuze nephroblastoma, nephro means kidney. Kwa hiyo blastoma no kwako ni tumor of the kidney ariko igaragara histologically nkaho ari ari kidney yo muri embryo. Umwana ukiri embryo utaragera fetus. Kidney ye uko ibisa niko icyo tumor ibira isa. So bivuga ko na glioblastoma ni tumor of the brain, astrocytes, but which appears like the glial cells within an embryo. We have a retinoblastoma. It means the cells historically look like the retina cells within an embryo. So these are embryonic tumors, if you can say so. Embryonal type of tumors, we use this suffix blastoma. Ariko nivuga ko ziba mu bana bakuru barazigira. So we have a lymphoma. We have a melanoma. So, okay, izi mbi zisimbutse nzigaruka nzagarukaho munyibutse. So rhabdo means striated. Myo means muscle. Sarcoma is a cancer. So this is a cancer arising from a striated muscle. So striated muscle, it can be skeletal muscle, is the muscles za biceps n'ibindi, cyangwa zikaba biriya y'umutima kubona ni striated. Leo means smooth. Myo muscles, and the sarcoma, this is a cancer arising from smooth muscles. Smooth muscles, you know where they are. Zifite muri tunica media of the vessels. We have them in the uterus. We have them in the wall of the gastrointestinal tract, from the lower part of esophagus, the stomach, the small intestines, and the colon. So iri ahantu henshi. Fibrosarcoma have seen arising from fibroblasts. So ngira ngo aho ngaho twabisoreza no tukajya muri ibyo bita exceptions. Exception ya mbere ni icyo bita misnomer. Misnomer, MISNNOMARS, misnomers. So misnomers ni ukuvuga ni ukuvuga ikintu kitari cyo, ni ijambo ukunda gukoresha. So when you say misleading and so on. So mis and noma, it means a name which is not appropriate. So na amazina dukoresha akoreshwa atyo, ariko nubwo bimeze gutya akoreshwa mu buryo butari appropriate ukurikije na histology. Bishaka kuvuga ikirere. Here we have tumors. Ubonye lymphoma wavuga uti this is a benign tumor arising from lymphocyte, ariko ntabwo ari. This is a cancer. Niyo mpamvu rero ari misnomer. Ni ukwera uburyo yandikwa ni nk'ari benign tumor, ariko it is a tumor, a cancer. Melanoma wavuga ngo it is a benign tumor arising from melanocyte, but it is not the case. These are cancers, very aggressive cancers. So nyagushize nabivuze ura muntu wa Robin wanditse biriya bitabo dukoresha cyane, ni nawe yishwe na melanoma yo mwijisho. So these are misnomers. So if I ask you to give examples of misnomers in the tumors, so these are good examples. Lymphoma, kubera ko normally yagomba kwitwa lymphosarcoma kubera ko it is not an epithelial cell. So yari kwitwa lymphosarcoma, ni yari kwita melanocarcinoma kubera ko melanocytes they are in the category of epithelial cells. So yari kwitwa melanocarcinoma ariko ntibikoreshwa. So therefore muzamurabona mu bitabo bya kera bashyiragaho ijambo malignant imbere, malignant lymphoma, malignant melanoma. So nimuzabona ibyo bitabo muzamenye impamvu iby'ubu babikuyeho. Kubera iki inama ziba zavuze ese ubundi dushyiraho malignant melanoma ko hatabaho benign melanoma, nuko cyitwa ijambo malignant, niyo mpamvu baje kurikura. So nyango muri pathology tugira icyo twita International Academy of Pathology, ihuza abapathologists bose ku isi. Dukunda kugira ko congress rimwe mu myaka ibiri, So ubu yabaye mu byumweru 2 bishize abereye muri Australia. So me, I presented what I had to present. I presented virtually. Ntabwo nagiyeyo, but each 2 years we have them. So okay, so iyo duhuye haba harashyizwe teams by WHO, World Health Organization, hariho teams yo kwiga kuri research, ibintu byanditswe muri research, byakozweho research mu minsi itambutse, bakabyigaho bakaba bati dukora iki kuri ibi, iyi ni indwara nshyashya cyangwa se ubu ni indwara niyo cyangwa ntabwo ari. So iyo duteranye batubwira hari indwara twakuyeho, iyi ngi ntimuzongere kuyitangaza diagnosis, ahubwo ibintu bizayo mubona bisa gutya mugomba kubyita gutya, none mubishyira mu yindi category. So ahora niwa inama ziterana zikaba bati okay, guhera ubu ntabwo tuzongera kwandika malignant lymphoma, kwandika lymphoma gusa, because there is no benign tumor arising from lymphocyte. It doesn't exist. So therefore we don't have a benign lymphoma, and therefore we cannot say malignant lymphoma if there is no benign lymphoma. So from now we will use lymphoma only, not adding that. So the same for melanoma, and the same from seminoma. This is a tumor arising from seminiferous tubules, of course in the testes. So uko yanditse gutya bavuga ngo ni benign, but this is malignant. It is a cancer, aggressive one. Okay, I can say fortunately these are treatable. Even in our country they treat them bagakira. So teratoma ni the exception. When you say teratoma, it is not enough. So teratoma ushize nabivuzeho iva hehe, hari amaserire stem cells aguma mu mubiri hirya no hino. Mu gihe runaka amaserire ashobora kwidifferencia niba if it is a cell which was had a predilection to become a skin. Niba iri muri ovary n'ubundi zakora skin, igira ubwoya, salivary gland, nako ziriya sebaceous glands usanga zirimo, imisatsi izemo, niba iri kuvamo amenyo yasangamo, ni ibintu tubona hafi buri munsi. So these sorry, is there a question?
Yes.
Which one? Okay, so teratomas, they are dense tumors arising from dense totipotent cells which have not differentiated in due time during embryogenesis, and they differentiate after embryogenesis, and they give rise to different kinds of cells. So uzasanga buri ifite the cells from ectoderm, endoderm, and mesoderm at the same time. So nizo bita teratoma. So then when it is benign, akenshi twongera ijambo cystic. When it is a cancer, we add, okay, when it is benign, we add a word mature teratoma. When it is a cancer, we add a word immature. Immature means the cells are not mature, they behave as cancer. Zirahari hano mu Rwanda kandi zikunda gufata abana, umwana kugira n'indi muri Dr. Yakane Butare afite imyaka 13. So she came bavuga ngo afite abdomen, acute abdomen, kumwe intestines zazifunze, no stools, no gas. But when I palpated, I feel there is a mass. So I called the surgeon. I was on it at that time. So then they programmed that because this was emergency. When it's stools and the gas is not passing, it is an emergency. So they programmed to do surgery in the morning. So when we opened, I was assisting the surgeon. So when we opened, we found it is a tumor of the ovary, hemorrhagic. It has destroyed the intestines. It has destroyed the urinary bladder. Many organs were affected, and it was so hemorrhagic that it was very difficult to do hemostasis. Then the surgeon called the most senior surgeon. He was a German surgeon. Then he came and he said, so I think you cannot proceed because the tumor has too much advanced. Maybe you can transfuse because the patient has lost a lot of blood because of the hemorrhage. You can transfuse, but you cannot remove the tumor because if it has gone into the liver, into the many organs, so you cannot exercise on the tumor. So you just close as per protocol. So it is inoperable tumor. You just close because of both these surgeons were foreigners. They didn't speak Kinyarwanda, so they told me to tell the mother of that girl what we saw. So I explained, and then I said, so the surgeon said, because of what we saw, this child, of course, will not survive, and probably she will not come out of the anesthesia. She may die. She may die while atarakanguka, or ashobora kuza gukanguka ariko ntabwo ari bumare amasaha menshi kubera ko so umudamu arabyumva ararira, nyuma aradushimira nyine ko tumubwiye ko bimeze. So umwana aza gukanguka nka saa 8 ambwira nshaka amazi, byumvikana yari ari muri shock, nshaka amazi, mfite inyota nyinshi. Byumvikana umuntu wabazwe ntabwo umuha ibyo kunywa cyangwa ibyo kurya. Ndamubwira amazi turi kuyaguha muri serumu, ihangane, nariya gusa ntabwo dushobora kuguha mu kanwa kandi wabazwe. Ariko I knew there is an intestine zasaga naho zitagihari, byose yarashwanyaguritse. So of course yamaze nk'iminota 20 na mama we, then rapfa. Ariko histology showed it was that immature teratoma, and she was 13 years, I remember. So teratoma can be immature, which is malignant, or mature, which is benign. Seminoma, it is always a cancer. It is a cancer, and this is a misnomer because when I repeat seminocarcinoma, because these cells look like epithelial cells, but it is not. So these are the misnomers. So hari iziva mu maraso. Okay, muri lymph node ni lymphoma cyangwa lymphocyte in general. Lymphoma ntabwo iva muri lymph node gusa, ishobora kuva no muri lymphocyte aho ziri hose. Hari niyo tubona muri hariho hose, no mu kuruhu zirahaza, and so on. So we have many types. Hari yitwa Hodgkin, yavumbuwe n'umuntu witwa Hodgkin, Dennis Hodgkin. Hari izindi zitwa non-Hodgkin. So iyo ari cancer iri liquide noneho y'amazi igenda mu maraso, niyo bita leukemia. Uzabona ngo umuntu afite leukemia. So leukemia, it means the cancer cells but which are saturated, they are not forming a solid mass, but they are saturating throughout the blood. So ni bayita leukemia. Ariko aya masere murabizi nyine akorerwa muri bone marrow. That's why, so the origin is the bone marrow. So the central nervous system, they are called glioma in general. Ariko iyo are benign with astrocytoma, ukongeraho subtypes when ibyo bireba pathologists, or when you be neurosurgeons, there's a subtypes. And when it is malignant, kwahise glioblastoma. Mbere bo bahitaga glioblastoma multiforme, ariko vuba ahangaha iryo jambo multiforme ryakorewe. So mu bitabo bya WHO dufite ibishyashya ntabwo bakiryongera. Salivary glands, hari indi tumor which is an exception, pleomorphic adenoma. Pleo, it means many, morph, it is shapes. So iyi nayo ni nka fibroadenoma in the breast. Niyo uyirebye birasa, uretse iyo uzi organ ni fibroadenoma cyangwa ni pleomorphic adenoma. Otherwise birasa. Pleomorphic adenoma niyo muri salivary gland, fibroadenoma niyo muri breast. Ariko histologically ziba zisa, ni epithelial cells and fibroblast proliferation both. So ngira ngo hanga ariko hari ikosa, iyi ntabwo agomba kuza hano. This must be in benign. It is benign. Gusa hari icyo tugiramo, hari ubwo hinduka cancer, icyo tuyita carcinoma X pleomorphic adenoma, meaning carcinoma arising from a pleomorphic adenoma. So here I will make a correction. So mesothelium, you know, this is a tissue. This is the peritoneum, cyangwa pleura. They are lined by a kind of epithelial cells called mesothelial cells. So these mesothelio cells, they can give rise to benign tumors, which would be benign mesothelioma, or a cancer which will be called malignant mesothelioma. So ahangaha ntabwo ijambo mesotheliocarcinoma ntabwo rikoreshwa. Ho byanze kunzongera ijambo malignant to show if it is cancer, or benign show that it is not a cancer. So mature teratoma na immature teratoma nabivuze. So I just explained in general how we give names to the tumors. Then I have shown you the misnomers, tumors which are given names which are not appropriate. Usually these are malignant tumors, but their names appear as if the tumors are benign. So there is a third category now. Ni tumors zihabwa amazina y'abantu bazibonye bwa mbere cyangwa bazanditseho bwa mbere.
Question.
Yes.
Excuse me, lecture. About those glioma from central nervous system. It is not well explained because we have even seen them later about astrostoma, but I don't get it well.
I have explained that glioma is a general term for the tumors arising from the astrocyte. So it is a general term, but as a pathologist for us, we don't give this diagnosis because we must give a diagnosis with a very well-defined type of the tumor. So when they are benign, dukoresha ijambo astrocytoma. So you understand it is benign tumor from astrocyte. When it is malignant, we call it glioblastoma. But glioblastoma and astrocytoma, both, they are globally called gliomas in the genome. So I don't maybe get where your question. Glioma are arising from astrocyte. If benign, we keep the name. Okay, we keep the name astrocytoma, but harandi magambo twongeraho. When, for example, it is very low grade, twongeraho pyrocytic. When it is somehow more advanced in grade, we call it diffuse. Iyo iri advanced cyane yenda kuba cancer tuyita anaplastic astrocytoma. So niyo wenda mukeme ntashatse kuvuga mbere hazamo n'izindi nka gemitocystic and so on. But all these are benign. Anaplastic is the intermediate between benign and malignant, and then glioblastoma, it is malignant. So I was explaining
that as I'm a beginner in pathology, I have to keep in mind that glioblastoma is malignant, while astrostoma is benign.
Yes, like that.
Okay, thank you.
So I was then explaining that there are tumors which keep the names of the persons who described them first. So niyo mpamvu hano mwabonye nsimbuka iri jambo Williams tumor. Williams tumor, it is a tumor arising from the kidney, ifata abana cyane cyane. So ubundi yitwa nephroblastoma. I explained the nephro just refers to kidney. Umuzuka harimo na nephrons ziriya the filtration. Glomerular filtration, nephrons. So nephro kidney blastoma, it is embryonal tumor. Nasobanuye cyane embryonal tumors. So histogenically yitwa nephroblastoma. Muri literature, mu bitabo, muri articles, bashaka gukoresha nephroblastoma cyangwa bagakoresha Williams tumor. So byose barabikoresha. And it is very frequent in Rwanda. And as I was saying last weekend, I was we were discussing issues with that professor from California. He was saying he sees many nephroblastoma Williams tumor in Rwanda, but in their country he doesn't. So there may be reasons we don't know. Maybe through research we will know. So this is okay. Now icyo niba gihe cy'ingenzi nuko babyita eponyms. Eponyms means the names given to a disease because of someone who discovered that disease. So Williams tumor, it is the eponym because it bears the name of someone called Williams who described this disease for the first time. The Hodgkin lymphoma, ahangaha nabyo nuko, Hodgkin's lymphoma, it was described for the first time by Hodgkin. So that's why it keeps this name. So twabonye nomenclature ni general, tubona misnomers, none tubonye niyo za eponyms. So hari izindi nyinshi cyane. Hari Bucket lymphoma. It is a kind of lymphoma y'umuntu witwa Dennis Bucket, yari mu so he was a British surgeon, so yaje kuvura kuri abaza muri za mission z'amadini, yaje kuvura mu Lago Hospital Makerere, Uganda. So mu mwaka w'igihumbi 1958 aza kubona hari utwana twinshi tuba dufite ibibyimba ku matama binini cyane kandi bikura vuba vuba. Arandika ati nabonye I saw a sarcoma in African children which is very aggressive. So arayandikaho ni aritika mushaka kubona kuri internet. So muri uwo mwaka nibwo yanditse. So abandi babibonye biwabo n'abandi baravuga ati no, we must know what is that. So they told him to take samples. So afata samples n'ubu twohereza samples muri research cyangwa ari n'umurwayi rimwe na rimwe azohereza mu Bwongereza. So haza kuba ikibazo, indege yagomba kuzitwara irazitwara ariko ntiyazigezayo, zirabura zimara iminsi 71 zitaragera aho zigomba kugera. So zigezeyo samples barazibonanye zaratinze, ariko basanga hari ibintu bidasanzwe birimo, harimo ibintu bimeze nko kubira nkaho ari infection aho kuba tumor. Bise kuya biterimo wenda iwacu kuya bavuga bataranzoga zikabira n'ibintu byose. So aravuga no, this must be an infection instead of a tumor. So what they did, now they contacted the microbiologists and so on. They say no, we must do culture, tissue culture, nkuko bakora culture ya urine bashaka infection. So when they did culture, they discovered there were viruses. So abantu bari muri laboratory ya testinze izo bakabonamo viruses. Umwe yitwaga Epstein, yari umuyobozi wako karabo laboratoire. Undi yitwaga Bar, Yvonne Bar, yari umukobwa mudamu. Undi yitwaga Acheng. So nyirango wenda mwamwarizo virology mwahise mwumva ko hariho habonetse virusi yitwa Epstein-Barr virus. Epstein ni wo wayoboraga iyo labo, Barr ni wo wundi bakoranaga, Acheng ntibamushyizemo mu izina. So hasobanurwa byinshi, abanyafurika bo bakunda kuvuga nuko yari umwirabura yari afite amaraso abirabura, biragaragara ku mafoto yo babonye. Ariko ibyo ntabwo twabitindaho. Amazina bahise iyo virusi yahise ihabwa amazina ya babiri, umwe arasigara. Biba n'inshuro ya mbere byari bimenyekanye ko hari kansa iterwa na infection. Ubwo bwa mbere bisa naho byamenyekanye mu bantu, ubundi byavugwa ku nyamaswa. Hari n'abandi babyanditseho mbere. Hari umudamu wabyanditse witwa Ellen White, we yabyanditse atari yize n'ubuvuzi, atanize amashuri menshi, yabyandikaho mu bijyanye na religion. Avuga ati ibiryo murya cyane cyane inyama murya zirimo germs, cyangwa se germs wabyumva n'ibintu bya infectious zishobora gukurura cancer. So ntawe bitinze icyo gihe, yanditse mu myaka y'1880, ariko nyuma ababaza kubibona gutya. So nuko bareba na za cells of the tumor basanga ni lymphocytes. Birumvikana we yabonaga ari sarcoma kubera yabonaga epithelial cells mwitama zo ziguma kuba ziri normal, nta gisebe kiba gihari, ubwo tumor ibirimo imbere. So basanga ni lymphocyte, so bayita Bucket lymphoma because the tumor was arising from lymphocytes, and the one who described the first time is called the Bucket. So yitwa Bucket lymphoma. So ni lymphoma zigaragara cyane na hano mu bihugu birimo malaria. My doctoral project I worked on that to kureba n'izindi lymphomas and leukemias which are not Bucket lymphomas but which may be associated with EBV twazibonyemo EBV, Epstein-Barr virus muri lymphoma zindi, ariko ntitwazibonye muri cancer ziva muri neutrophils and so on. So that was bucket lymphoma. So hari research zakomeje gukorwaho abanyasuwede bavumbura molecular, harimo translocation chromosome y'umunani na 14, abafaransa nabo bafite ibindi babikozeho cyane. So yakozweho research cyane ngira ngo irazwi. Gusa this is the most aggressive cancer. Birazwi ko ariyo cancer ikura vuba. Muri literature muzabona bavuga ko mu masaha 28 ishobora kuba yikubye inshuro ebyiri uko yanganaga. Mu gihe izindi ushobora no kumara irakura ariko ishobora kwidubla uko yanganaga nyuma y'umwaka cyangwa igihe kinini, ariko iyo amasaha 28. Gusa mu gukura kwayo bisobanura ko kanseri ubundi ikura vuba vuba, inyinshi ntabwo ari zose zikura vuba vuba, ishobora kuvurwa vuba igakira. Mpamvu bucket lymphoma no mu Rwanda bayivura umuntu agakira burundu, umwana agakira burundu. So we have many other tumors, muzabona kuri liste ntabwo twazitindaho, in terms of eponyms. So if there's a question, you can ask it, otherwise you can take a break. Any question? We have seen in nomenclature in general, we have seen misnomers, and we have seen eponyms, the concept of eponyms, and then nabiriya bya embryonal tumors bisa naho ari category uko ifite blastoma as a suffix, banazibuza nephroblastoma, retinoblastoma, glioblastoma, and so on. So otherwise uko tuzajya muri systemic pathology, nitugera kuri hematopoietic cancers, nuko tuzagaruka ku bintu by'amalymphoma na leukemias na multiple myeloma, it arises from plasma cells. So this is, we must also take it. Multiple myeloma arises from plasma cells. So we will be coming back. If we go to the tumors of urinary tract, we will see we remove the tumors, we will come back. No kureba zifata abantu bangana gute clinically, how you can suspect it. So all this you see in systemic pathology. Because in systemic pathology, we apply principles we are learning here in general pathology to each organ to describe the diseases which can be seen there. So it seems there is no question, or you can ask it after the break. So can you have a 10 minutes break? Okay, welcome again. I hope you can hear me and see the screen. Do you see the screen and hear me?
Yes.
Okay, thank you. And I saw that there was a question in the chat. So when I'm presenting, because I do a full screen, I don't see the chat. So it is okay to write the question there, but if it is a burning question needing it to be answered, better to just talk. So now we go to the part of carcinogenesis. So this term, when you see it, you can think that it is only for carcinomas because there is a word carcino, but carcinogenesis just means the process of cancer development in general. It can be a carcinoma or not a carcinoma. In order to understand this, we have to understand very well the principles of cell signaling, as you have seen in molecular biology and cell biology. You know that for when there is an information which must be sent to a cell, that information is sent via what we call a signaling molecule. Usually, signaling molecule can be a growth factor or growth inhibitor. So it brings the information. This can be a protein, it can be a hormone or another thing. So it will be received by a receptor which is on the cell membrane, and sometimes the receptor can be within the nuclei. And you must know that, for example, the receptor for estrogen and the progesterone, the receptors exist in the nucleus, not on the cell membrane or plasma membrane. So the receptor receives the information and sends the information to the nucleus, because the nucleus is the government of a cell. So is it like if someone is working on the border, customs, for example, on the border, if you receive an information that needs for which a decision, a general decision must be taken by the central government, you will send the information to the central government. Therefore, this is the receptor what it does. So it means the signaling molecule will not enter into the cell, no. Except the estrogen, it will enter, but it will not directly exert its changes. It must be received. The information must be received by the receptor. The receptor send the information where it will be processed. So here the signaling molecule will not enter as such, but depending on that molecule, the receptor now will generate an appropriate information, which will be sent to the nucleus. How is that sent? There is transduction now. Transduction, it means the receptor processes that information and generates a new information or an adapted information, which can be now interpreted by the nucleus. And this process here, it is called transduction, and it is done by what we call second messengers. So second messengers ni nk'intumwa zaba ziva kuri border zijya muri kapitale zijyanye amakuru. So the second messengers, they will go and activate the cellular response either by transcription. Transcription, it means the DNA will make copies of messenger RNA, and of course, from messenger RNA, we have proteins. And another possibility, it is to activate the cell cycle, to activate the cell division. So therefore, here you must understand that there are 5 steps. There is a signaling molecule which can be growth factor or growth inhibitor. There is its receptor, that is 2. 3, second messengers or translation. 4, we have transcription. 5, we have cell cycle. I hope you understand or you remember this because you have covered that in molecular biology. Now, why are we bringing this here? First of all, I said that there is no cancer without mutation. A mutation means there is the abnormal change within the DNA. In humans, of course, kubera ko DNA tugira niyo itanga ibika amakuru ya genome. Hari viruses zifite RNA gusa zitagira DNA, so nazo ushobora kugira mutation in RNA. But here we are talking about DNA because we are talking about humans. So a change in the DNA then can appear where that mutation can appear to a gene which produces the signaling molecule. Bivuga ko ishobora no kuba atari kuri iyi serire yakira molecule, ahubwo change zarabaye kuri serire ikora iyi signaling molecule. That is one. Niho muzamubona muri cece tuvuga ngo uyu muntu afite amplification of the growth factor runaka. Or this can be normal, but a gene which produces the receptor here is abnormal. So, for example, in our country, or so is someone opening the mic? Please keep your microphones muted if you are not talking. So I was giving an example of problems with receptors. Globally and here in Rwanda at East Africa, we have molecules we test. We have a test we do to know if a breast cancer has a mutation in the gene called the human epidermal growth factor receptor. So this is the kind. So nko muri abo ngabo iyo dusanze afite izo receptor yabaye amplified, nuko receptor iziba zabaye nyinshi, ubundi yagombye kuba imwe. Niba rero umuntu azajya amakuru ari umwe yakiriwe n'abantu benshi, ba bantu buri wese arohereza muri central government ukwe, ubwo abo bantu n'aba receptors ndi kuvuga barohereza muri central government buri wese amakuru, birajya muri central government bisa naho n'ubundi haje inkuru nyinshi, haje signaling molecule nyinshi ariko atari. So ibyo byose bitera ikibazo, bituma nucleus ireagisa mu buryo bukomeye, ikahita isaba ko cell division yihuse irimo n'amakosa ntakosorwe and so on. So ibyo rero bituma amplification itera ikibazo. Hari nicyo bita overexpression. Overexpression ntabwo copies za receptor zabaye nyinshi, ahubwo ubundi normale nkuko mwabye iyo receptor yakiriwe amakuru ikayatanga kuri second messenger, ihita igaruka muri status twakwita normal, reka twite normal cyangwa se non activated status. Nirya rero haba icyo bita overexpression. Ni igihe umuntu ni igihe receptor yakiriye amakuru yamara kuyatanga igakomeza ikitwara nkaho yakiriye andi makuru. So it means a signaling molecule came once, the receptor received the information and sent it to the nucleus, but it continues to be activated. So being still activated means it sends again other second messengers. So the nucleus will receive a lot of information as if there are other new signaling molecules. So that is the overexpression. Naho amplification ni igihe gene yabaye nini. So icyo urabona niyo ukoze sequencing of ya gene usanga usanga yagize copies nyinshi, usanga gene yabaye ndende kurusha uko yagomba kugenda. So a mutation can occur to the gene which is responsible for producing the second messengers. Ariko murabizi ko second messenger ubundi ntabwo zigenda gusa, hano haba icyo bita pathway hano ebyiri zizwi cyane, hariyo bita tyrosine kinase n'inda GTP coupled proteins. So nuvuga iki kintu receptor kiri attached ku bindi bintu hano aribyo bishobora kuba tyrosine kinase cyangwa se GTP coupled proteins. Izo n'ubundi nizo zactive noneho second messengers zikagenda. So therefore there can be mutations in the genes producing the proteins here, or mutation in the genes producing the second messengers themselves. Ikindi gishoboka nuko byose bishobora kuba ari normal, umuntu akagira mutation in the genes which control transcription. because our DNA doesn't just decide I can do transcription nk'umuntu ubyuka bati uyu munsi nshaka kujya guhinga cyangwa kwiga cyangwa nkabireka. No, DNA is controlled. It cannot undergo a transcription if not allowed. So it is allowed by genes like a P53 cyangwa n'izindi genes, iyo bita retinoblastoma gene 1, Williams tumor 1 n'izindi nyinshi. So izo genes once they are mutated ntabwo zizakora akazi kazo. They are like auditors. Niba ufite auditor uzakora ku ikigo kikora neza batiyoboye umutungo wa leta, noneho auditor ubwo akaba ari corrupted, ntumvikana, noneho n'abandi bacunga ibintu bazakora ibyo biboneye baziba bazagira bate. So niba izo genes zishinzwe kwemera sere gukora transcription ziri mutated, it means they are corrupted. Then there will be no break, there will be no check of the DNA. Izo genes icyo zashinzwe no kwanga ese DNA ko igiye kuba transcribed, ubundi ntabwo ifitamo nk'amakosa. Niba ifitemo amakosa mu mutation ntabwo yemererwa kujya muri transcription cyangwa kujya muri cell division, nabyo ni kimwe. So hari ibyo mwabonye bita checkpoints hagati wenda ya G1 phase na S phase and so on. Nicyo izo checkpoints zikora. Haba hari gene ishinzwe kubanza kako nta kosa na rimwe ririmo muri DNA mbere yuko yemererwa gukorwa indi copy yayo cyangwa gukora transcription cyangwa habaho cell division. So iyo izo checkpoints zifite ikibazo, genes zishinzwe ziri corrupted, ziri mutated, then that control will not happen. Therefore the cell will be allowed to undergo mitosis kandi ifite mutation. So normally when these genes are there and are working very well, when they find there is a mistake, there is a mutation, they command genes responsible for repairing. And these genes responsible for repair, they will do their task. When the genes which control the division or transcription find that the repair cannot happen or the repair failed, then they command the cell to be killed by apoptosis. Nibyo P53 as a gene ikora, nicyo kintu ikora, hari n'izindi zifasha ariko niyo cyane cyane. So izo rero nazo zishobora kugira ikibazo, ni ukuvuga we may have mutations in the gene responsible for transcription or the gene responsible for controlling cell division. So murabyumva rero, iyo umuntu afite cancer ashobora kugira mutation at any level here. Kandi iyi mutation iyo umenya aho iri nibwo ushobora guhabwa imiti. So nk'urugero hari umuti, hari zirano za kanseri zo mu maraso, leukemia, yo ita chronic myeloid leukemia, ikunda kugira ikibazo muri iyo tyrosine kinase hano muri iyi transcription. So iba ifite ikibazo over expression. So kubera ibyo, iyo tubipimye tugasanga niyo afite, hari umuti uzwi uwo muntu ahabwa witwa Gruvec, nano mu Rwanda urahari. Abo bantu urabafasha cyane. Before we are not doing that, ariko we developed the research project, SHK irayifinansa amafaranga, ntabwo yari menshi ariko at least we have shown that it is possible. Dukoresheje machine zihari zisanzwe zipima ibindi, dushobora gupima ayo magene twamenye ko umuntu afite icyo kibazo muri tyrosine kinase, therefore tugasaba ko ahabwa uwo muti. Ikindi, iyo agiye ku muti agomba gukurikiranwa buri mezi 3-6 gutyo, twasanga noneho amaserire ya kanseri akiri menshi, tugasanga n'iyi mutation iracyarimo, icyo gihe tumenya ko wa muti utari kumufasha, tugasaba ko bamuha indi classe ikomeye kurusha. So byose ni evidence based. So therefore ibintu tuvuga hano ntabwo ari birangira muri theory, ahubwo nibyo byagiye bituma abantu batekereza bati, if this cancer, for example that breast cancer, iterwa na over expression ya human epidermal growth factor receptor hanga, ni gute twazibira iyi receptor ntikomeze kuba upregulated, donc ikomeza kuba expressed. So hari umuti witwa trastuzumab ubikora. ubihagarika iyi ntikomeza kuba over expressed. So kuba ufite breast cancer nabyo turabipima by immunohistochemistry. On every breast cancer case we diagnose, turabikora. So ibitaro mu Rwanda bikora ntabwo ari byinshi, na ni CHK, Kanombe, Faisal na Butaro, but it is done almost for every patient. Ariko ikibazo kikaba kimwe, iyo abaye positive, iyo dusanze afite iyo mutation bigenda bite. So uwo muti trastuzumab urahari ariko urahenda. So kire y'amezi nyango 18 heruka ishobora gutwara amafaranga miliyoni 70. Mituweli zitayishyura, so cyari ni ikibazo gikomeye, ariko ubu hari ibyatangiye ku leta yagerageje gushaka uburyo imiti yaboneka ku giciro gihendutse kurushaho, ivugana na ba nyir'inganda and so on. So all these things they have implication. Ariko icyo nashaka kwerekana uko buri cancer habamo mutation at these 5 levels. Turabigarukaho mu yindi presentation which is about the molecular basis of cancer. So it will be a separate presentation. So akanga kana introduction. So it is the same thing. We have a receptor, okay, growth, a factor which can be growth on inhibitory. Then it's receptor, effector nizazi zohereza second messenger ikajya muri DNA, muri nucleus, igateka DNA synthesis and RNA synthesis, which is a transcription or activating cell division from cell cycle. So what is carcinogenesis now? Carcinogenesis is the pathomechanism of development of neoplasia and is the result of non-lethal mutations of genes controlling cell growth and division. I think they understand the mechanism of development of neoplasia. So it can be cancer or benign neoplasia, as we have seen. But most importantly, there are always mutations which are non-lethal. Lethal ni ikintu cyica. So iyo mutation itumye serire ipfa, ibyo biba ari amahire ku muntu, kubera ko nyine bita ikibazo cy'ikimo. Ariko iyo mutation ibayeho serire ntipfe, ya serire ikemererwa gukora indi division. Therefore hakabaho serire zirenze imwe zifite ya mutation. Cancer ubwo iba yatangiye. Unless there is a very strong immunity which will come and kill these cells. Ariko akenshi ntibiba bigishobotse iyo when the cell which has a mutation is allowed to undergo mitosis. It is now the cancer, iyo serire ya mbere bayita transformed cells which has a mutation. When it is allowed to undergo mutation or cell division, now problems arise. Cancer will develop. So that's why that gene, for example, P53, when it finds that a cell has a mutation, it makes everything possible in order to repair that mutation. If the mutation cannot be repaired, then it commands the killing of that cell by apoptosis. Now, the problem is when it is not possible to repair and it is not possible to kill that cell. So, meaning that that mutation becomes non-lethal because the cell is not killed and it is transmitted to the daughter cells. So the natural history of most cancers can be divided into four phases, like malignant changes into the target cell, which is the transformation, mutation, growth of the transformed cells. And I think I have said last time, when we are talking about tumors, when we say growth of cells, so it means this cell which has a mutation or is transformed, it undergoes mitosis. This is growth. Then there will be local invasion of surrounding tissues. We have seen in the pictures, and the distant metastasis. So metastasis in fact cancer if uduce duke tukinjira muri blood vessel cyangwa lymphatics cyangwa mu zindi nzira tukajya muri distant organ. So mu zindi nzira ni nyinshi, hari ibinyura nk'ahantu hafunguye, for example a cancer of the ovary can go through the abdomen kubera ko ni espace ikagenda ikajya nko kuri stomach cyangwa ikava kuri stomach ikajya kuri ovary. Ni muri za eponyms navugaga bayita Krukenberg tumors. A cancer of the ovary going to affect the the stomach, because both of these organs are intraperitoneal organs. So between them, there is just a space. There are, of course, intestines, but it is an open space. So hari na cancer cells, there are cancer cells which go through nerves. For example, in a prostate cancer, when we diagnose it, we usually don't look for invasion of the lymphatics or blood vessels. We see if there are cells around nerves, nerve endings or nerves. When you see that, it is a sign that probably cancer has gone far, just following the nerves. Bibaho no muri one type of cancer in the salivary gland called acidic carcinoma. Okay, yes, we shall see that in a systemic pathology. So the question is, how long does it take to produce an overt tumor mass? So here we not give how long in terms of time, but in terms of how many cell duplications. So you know that an epithelial cell, for example, is around 10 micrometer diameter. So when this cell does 30 population doublings, is doubling is exponential. So it produces, this is 1 billion cells, and 1 billion cells weigh about 1 gram. You understand, that's very small, around 1 centimeter diameter. And this is a smallest clinically detectable mass. Na keretse iri ku ruhu, otherwise iri buri mu gifu cyangwa ahandi, nibwo igeze ahangaha nibwo umuntu ashobora kumva ko ari ikintu kidasanzwe. Iyo ari gato cyane ntacyo wumva. No muri breast ntacyo wumva. Iyo kamaze kungana hafi santimetero imwe nibwo umurwayi akumva cyangwa nibwo kagira signs and symptoms. So when there are 10 further doublings, we have now 10 power of 12 cells, and these cells, they weigh about 1 kilogram. And 1 kilogram they say it is the maximum size compatible with life, but it depends, of course. Because when umuntu ntabasha kugira 1 kilo mu mutwe, no bigera ntibishoboka aba yapfuye mbere, because of the way it is. Ariko murashaka kukigera muri abdomen, tubibona afite a cancer of 2 kilos or 1.5 and so on. So but this is in general. So the maximum size compatible with life. So uko bigaragara ko yango cancer izagaragare, we must have at least 1 billion cells, cancer cells, in order for it to be seen by using the eye. So if we go back now to the cell signaling, what you have to retain is that when we start the general pathology, we say that in our body we have always a balance between promoting mechanisms and inhibiting mechanisms. So our genes, they fall into different categories. The first category, it is proto-oncogenes. These are genes which always promote the growth of cells. Growth, as I said, it means cell division or producing new cells. So these are called the proto-oncogenes. So they promote the growth of cells. The second category, it is called tumor suppressor genes. So as is because they have seen that these are the genes which help the body to fight against cancers or tumors. So you understand that what they do is opposite to the proto-oncogene. This is in a normal condition. So when now a gene, when a proto-oncogene has a change, has a mutation, it is no longer called proto-oncogene, it is called oncogene. So proto-oncogene is a normal, oncogene, it is mutated. So as I said, then the proto-oncogene are subdivided into 5 groups. Growth factors, you remember what we saw in the cell signaling, receptors, second messenger, transcription factor, and cell cycle promoters. And as I explained in that diagram, the mutation can appear at any level of these genes, the genes producing these factors. So on the other hand, we have a tumor suppressor genes, as I was explaining. So they follow the same. We have growth inhibitory factors, we have their receptors, we have their secondary messengers, we have transcription inhibitory factors and cell cycle promoter inhibitors. So meaning that they fight against the cell cycle promoters we saw in the proto-oncogenes. Now here we have a sixth group, which is this gene P53, which control that DNA is normal, and if it is not normal, it triggers the repair mechanisms. If the repair mechanisms fail, then it commands the killing of that cell by apoptosis. So that's why this gene is called the guardian of the genome. Ni nk'aho ishinzwe kurinda genome yacu ngo itazamo amakosa, because when there is a mistake, there is a mutation, it means if not repaired, cancer will come. So this doesn't have, of course, a counterpart in the proto-oncogenes. Otherwise, izi ngizi murabona ko ni counterpart of what we saw on this one. So this is how you must understand the carcinogenesis. So molecular pathogenesis of cancer, in summary, there are hundreds of cancer-associated genes. And some such as P53 are commonly mutated. Murabyumva, cancer nyinshi zifite mutated. Kubera ko niba yawe mutated, ibintu byose bizazamba. Ni nk'uko bavuga ngo umuntu ni ushinzwe kugenzura ibintu, ni umuvunyi cyangwa ni auditor general, nawe akaba ari corrupt. Birumvikana n'abashinzwe finance bazakora ibyo bashaka, ntawe uzabahagarika, niba n'abashinzwe gufata abanyabyaha nabo bari corrupted, ababyaha bazakomeza bakora ibyaha. So P53 is important, and unfortunately, of course, it is the most commonly mutated in any type of cancer. Others are affected only in certain cancers. So, and the implication is that ntabwo wavuga ngo reka ndebe uyu muntu nemeze ko afite kanseri ni ndeba P53. No, it is not possible, because P53 can be mutated in any type of cancer. But there are some mutations which are specific to some types of cancer. So iyo uyibonye muhita uvuga, okay, this person has this cancer because this mutation is only known to occur in such type of cancer. So consider cancer-related genes. So we must consider the cancer-related genes in the context of 7 fundamental changes in cell physiology that together dictate the cancer. And there's a 7 fundamental changes are these ones. First of all, in cancer, the cells have self-sufficiency in growth signal. Self-sufficiency, no kuvuga cell kugira ngo ibyara indi. Growth, you understand, growth ni ukubyara indi. Kugira ngo ibyara indi, iyo ubwayo, iyo cell iba yifitiye ubushobozi bwo kwishyiriraho itegeko, kandi itegeko rikubahirizwa ikahita ibyara indi. Kandi ubundi mu buryo busanzwe, selire akenshi ikontororwa n'izindi. Ariko this malignant cell, which this mutated cell has a self-sufficiency. It doesn't need the other cells to help it to undergo cell division. It has created a system whereby it can do anything. It is also insensitive to growth inhibitory signals. So kira cyambere no kuvuga ko ni ubundi ni oncogene zafasha, proto-oncogene zafasha. Kuri iki cya kabiri ni tumor suppressor genes zifite ikibazo. So this cell will be insensitive to tumor suppressor genes. Third, it will evade programmed cell death or apoptosis. Evade no kuvuga irayiceremba cyangwa se okay. Kundi umuntu avuga ngo yakarose amasomo. Ikora ku buryo ntaho ihurira na P53 kugira ngo ikosora amakosa, cyangwa igakora kuri ya P53 nayo iba corrupted. So this is the evasion of programmed cell death. So fourth, it has a limitless replicative potential. And I remember there was a question. Someone asked, how do you compare cell division in a cancer and cell division in normal tissue? For example, in the same organ. A cancer of the liver the cell division in the cancer of the liver and the cell division in normal cells of the liver. Umwe yaravuze ati I think in cancer selile zibyara vuba kurusha muri normal tissue, ariko ntabwo ari ukuri buri gihe. Bishobora kuba ariko akenshi ntabwo ariko bimeze. Ntabwo muri cancer selile zikura vuba cyangwa se zikora cell division vuba, ahubwo ikibazo nuko zidashyiramo break hagati. Donc hari ugukora cell division vuba ikayirangira kurya biva muri G0 cyangwa G1 bikagera muri M phase. Ntabwo rero muri cancer ako iyo etape ikorwa vuba cyane ukurikije na normal tissue, no. Ahubwo ikibaho muri cancer nuko irangije mitosis itejya muri G phase directly, mu gihe muri normal tissue hazamo pause hakamara igihe iruhutse iri nko muri G phase. So nibyo tandukaniro. So here you have a limitless replicative potential. So it means it is labile. It becomes labile. Nubwo yaba ari tissue itari labile, ibihinduka labile if it is a smooth muscle cell or skeletal muscle, which are normally permanent cells. They become labile cell. It doesn't break. It doesn't pause its cell cycle. There is a genomic instability caused by defects in DNA repair. So there are things maybe we'll see thereafter, but our body has many systems whereby it corrects the mutations. Nibyo bita DNA repair mechanisms. So now the problem in cancer is that they usually as a patient they have lost, or as it says, they have lost their DNA repair mechanisms. They are no longer working. If they are not working, then ni ukuvuga ko kandi na P53 iza yagomba kubona ko repair yananiranye ikica selile na iyo P53 ikaba itakiri gukora. Therefore za selile zizakomeza zibyarire izindi zibyare izifite ibibazo gusa za kanseri bitume irushaho gukura. Ni nabyo bituma habaho limitless replicative potential. So another thing is that there will be sustained vascularization, neoangiogenesis. I think this is also a hallmark of cancers because they acquire new blood vessels, and because of that they bleed very, very easily. You touch, there's a bleeding. And I know this is a sign that you usually describe, clinicians they describe. They say there is a tumor which is easily bleeding on a touch. So you touch on the cervix, it bleeds. Or in the stomach, you are doing an endoscopy. The endoscope touches a mass and it bleeds. So this is a sign that it is a cancer. And because the benign tumors, they don't acquire very much new blood vessels. They just have a few blood vessels. And last week on Wednesday, when I was teaching the postgraduate in surgery, in plastic surgery and pediatric surgery. So I asked them the question, so why do you think the cancer makes its own new blood vessels? What is the mechanism? So the answer is that mu mubiri wacu tugira gene ishinzwe kureba ko hari igice cy'umubiri kitari kujyanwa amaraso ahagije, hypoxia cyangwa oxygen ahagije amaraso ahagije. So this gene is called the hypoxia inducible gene. Or factor or gene, so the same, but the protein itself is a factor. Hypoxia induced factor HIF. So iyo rero amaserera ari gukura ya cancer kubera ko ibi kubyara amaserera bakanamara amaraso atari kuhagera ahita agira hypoxia. Nkibaza last Wednesday umwe yaransubije ngo it is because there will be an anaerobic respiration. Of course izaba ihari ariko anaerobic respiration ntabwo ari itera cancer, ntabwo ari ituma haza blood vessels. Ahubwo iyo factor hypoxia induced factor ikikora iyo yumvise ko hari hypoxia yo ubwayo ihita isaba the vascular endothelial growth factor zikaza ari nyinshi zigakora new branches of blood vessels. So this is just the simple mechanism the cancers use to make new blood vessels. And of course, you can understand that in the drugs or medicines which are produced, which are being manufactured to treat cancers, hari ubwo umuntu ashobora no gukora umuti ahubwo uzafasha izo serire kutumva ko zageze hypoxia. No kuvuga ukoze kuri seze zitumva ko hawe hypoxia, ntabwo ya HIF izinducinga new blood vessel formation. Ni hataza new blood vessels, ya amaseri ya kanseri yakoze amashyaya azabura oxygene apfe, and therefore cancer ye gukura cyangwa se igabanuka. So this is one of the mechanisms which are being exploited. Aba researchers explore akavuga ati ni gute twabigenza tukabuza ari amaserire gutuma habaho new blood vessel formation. Kuko nk'ubu ushoboye ku arwaye kanseri ukamenya ko hari blood vessel imwe ijya ari atari imwe ijya ukayifunga, nta kanseri izapfa. Ikibazo nuko ishobora gupfa none hari amasere yatambutse yanyuze muri veins and lymphatic yagiye ahandi. That's and you cannot know. But at least when you cut vascularization of a cancer, it is one of the methods of at least reducing its size. Then sixth, it has ability to invade and metastasize. So this I have explained. It infiltrates, it destroys basement membranes. Whatever it meets, it destroys. And it can enter into blood vessels, lymphatics around nerves, and metastasize to distant organs. So therefore, now we arrive to a diagnosis of cancer scientifically. So cancer is a genetic disorder caused by DNA mutation that are, for the most part, acquired spontaneously or induced by environmental inserts. So this mutation can be acquired spontaneously, gusa ari ikosa ryizanye muri DNA kuko iyo iri gukora replication ikashobora kuzamo ikosa, cyangwa se iryo kosa rikaba ryatewe n'ibintu biva hanze, environmental insert. And when we started, I said that 60% of the reasons of having a cancer are due to environment. 42 inherent genes changes. Hari a gene tuba twararazwe zifite utubazo ariko ntizibe expressed. So we will come back maybe in the afternoon on the molecular basis of cancers. Ariko icyo ntavuze nuko proto-oncogene, they are expressed in autosomal dominant way. This means that a mutation in a single allele is enough to be expressed. While the tumor suppressor genes, they are expressed in dominant auto in a recessive, autosomal recessive way. So it means for the mutation to be expressed, both alleles must be affected. So icyo mbivugiye ni iki, nuko rero nko ku tumor suppressor gene umuntu ashobora kugira a mutation in single allele because ari recessive, ntabwo izaba expressed, ntazagira tumor. But that mutation will be transmitted to the offspring, ku mwana we. Umwana we nawe ashobora kuba gutyo, ariko ashobora no kugira ibyago undi mubyeyi we wa kabiri nawe akaba yarafite that mutation which is not expressed. Byahurirana noneho we nta kindi izahita igaragara. Ariko icyo ni kimwe, iyo urumva ntacyo yanakoraho ngo ayirinde. Ariko kimwe na wa mubyeyi we n'uwo mwana niba afite mutation in one allele of the tumor suppressor gene, indi mutation to the second allele can be caused by environmental factors. Aho ngaho this is something we can prevent now. This is a definition given by in this book, Robin's Basic Pathology, the one I nakunze kuvuga. So what happens then in a diagram for a cancer to happen? Okay, I can catch one. Okay. So there is a transformed cell here. Transformed cell, as I explained, it is a cell which has a mutation. Okay, let's change the. Okay, this a cell which has a mutation is called a transformed cell. Umuntu ashobora kuyigira, ikibazo noneho nuko izemererwa kubyara izindi. Mu biyemerera mu bituma ibyara ibindi cyangwa mu byagiserera kugira ngo ibyare izindi, akenshi hazamo ibintu byo hanze, chronic infection, usanga afite mutation ariko muri cervix, ariko kubera arwara za syphilis zihoraho zira sexually transmitted diseases, imitezi, so ugasanga bitumye ya cancer cell ihawe uburyo bwo kwimultiply. So when it multiplies, it forms the primary tumor. So this is very important to understand. And in the primary tumor, you see that the cells, they look like this transformed cell, but there are here 4 cells which don't look like these ones. Ibyo bivuga ko muri cancer ntabwo cells zose ziba zisa, bitangira zisa ariko nyuma zigenda zihinduka. Why? There are many reasons. Muzabisoma, ariko icy'ingenzi icyambere kibitana nuko mutation steril ifite izi zayikomotseho hari izigeraho zikagira izindi mutations zitavanye kuri iyi zakomotseho, kubera bya bibazo twabonye hejuru. Repair mechanisms ntihari, bivuga ko a new mutation will not be repaired, so they acquire new mutation. So nubwo twavuze ko muri cancer and benign tumors they are monoclonal, noneho hazamo n'ibyo bita subclones. So mbere yari clone imwe, none hajemo subclones, niho bavuga ubwoko n'udusekuruza n'uduki twose. So then these new clones usually they have different behaviors as the primary ones. Nizo uzasangamo havukamo ishobora gushwanyaguza basement membrane. Izi za mbere ntizabishoboye, ariko iyi yo yabishoboye. Basement membrane here it is destroyed, and some of them they can even invade blood vessel and metastasize through blood vessel or lymphatic. Murabona ko iyi ni blood vessel berekanye hano. So when the cells detach from here, they enter the stroma, and then they destroy the wall of a vessel, usually the lymphatics cyangwa se vein. So personally, I have seen many cases of cancers, but ntayo nigeze mbona iri yaciye atari. So because maybe it is very tough, ifite za internal and external lamina, elastic lamina, ariko ikagira na a very tough muscle, maybe that is the reason. Ariko it is not enough. Maybe there may be some biochemical reasons. So then the cancer cells will enter into the blood stream, cyangwa se lymphatic. So igezemo ihurana na immune cells. Here we have a host lymphocyte. It can be natural killer cell. It can be any other monocyte and so on. Ubundi izi cells zirazirwanya zikazica. So there is a facility called the confocal microscopy. So we don't have it, of course. So where I did my doctoral studies yari hari, ariko ushobora ushyiramo sere za cancer ugashyiramo na natural cancer cyangwa T lymphocyte. So T lymphocyte ukayireba neza iri kwataka ayo masere ya kanseri, ukabonera yishe ubireba n'amaso. So our immunity is strong enough to kill most of these cells. The problems comes when the immunity is down, itself is down. Aha niho hava concept ivuga ngo icyo bazita immunotherapy, muzabibona muri kwiga oncology. Hari imiti baha umurwayi yita chemotherapy isanzwe, wenda yica amaseri bisanzwe, ariko hari n'indi yongera immunity, ikayiha imbaraga. Kongera immunity kenshi no kuvuga gutuma aya maseri lymphocyte zigira imbaraga zikaba zakwica cancer cells. So otherwise, the platelets will come and they aggregate around these tumor cells. Ibi bituma bikora tumor embolism, uko mu cyangwa thrombus. Tumor thrombus cyangwa embolus nuko byanditse aha. Yes?
About kuri iyo case yuko our immune zishobora kwica cancer cell, ko twize yuko kugira ngo immune yice any cell mu mubiri nuko iba yabaye recognized as antigen idahura nyine niyo mu mubiri, kandi izo cancer cells ziba ari za zindi zo mu mubiri n'ubundi twari dufitemo. Ubwo twavuga ko ari nka auto regression iba yabayeho wenda kuko zabaye multiplied ari nyinshi cyane. Mbega n'iki kindi usinga immune system kugira ngo imenye ko ari cancer cell?
Yeah, I understand your question. That is a very good question. And we discussed it with this postgraduate in plastic surgery last week. So there are two ways. First, when there is a cancer, there is formation of new antigens or new epitopes. Ngira ngo murabizi epitopes na antigen aho bitandukanye. So antigen iba ari nini, none akantu gato cyane ko kuri antigen which can be recognized by our immune system niko bita epitopes. So ishobora kudakora new antigens but it can form new epitopes. Because kuba ititwara nka no cells nuko ifite ikindi kintu kidasanzwe. So that thing can be recognized by our immune cells as an antigen or at least as an epitope. That is one way. So epitopes antigen will be recognized by antigen processing cells. Zizifate, zizipresente noneho kuri T cells and so on. You learn that in history in immunology. So ku ruhande rwa kabiri niki nuko cancer cells they will mask some of their antigens. No kuvuga cells zifite antigen bisanzwe ariko cancer niza izakora ku buryo ipfuka za antigens. Nizipfuka umubiri ufite systeme noneho ya natural killer cells. Natural killer cells zishinzwe iki? Zo zishinzwe kwica ikintu cyose zisanze kibura self. So tubita cells which are missing self. Cells which are missing self or self antigens. Yo isanze hari ikintu kiri aho ngaho kidafite ikiranga cyerekana ko ari cyo muri uwo mubiri. Birumvikana iyo antigen ziri masked ntabwo T cells zizazibona ko T cells recognize antigens, but natural killer cells will recognize cells which are missing cell. So meaning that niba cells ziri kubura antigens ni NK cells zizazifata, niba zagize new antigens ni T cells zizazifata no ku mubiri wacu wareba. I hope you understand.
Yes, thank you.
Okay, and it is a good question. It shows that you follow and you think it deeper. That's great. So is this ambulance then will arrive at a certain area noneho itangira ive muri blood vessel vuga ngo tugomba kuvamo tukigira kuba hanze hanga. So ibyo ubundi babyita homing. It means cancer cells, they will arrive at a certain site of our body and they say, okay, we have to leave the blood vessel or lymphatic and they go to stay here around. Now you can ask how these cells will recognize where they can be well received. Just until now, I don't know the answer and I have not seen in literature. Ariko ikizwi ni iki? Ikizwi nuko niba ari nka selile za cancer of the prostate byanze bikunze zigiye zishakisha bone. Ntabwo abantu bazi impamvu ariko ni observation. Iyo umuntu afite prostate cancer when it metastasizes ni gace izajya muri liver muri lung ahantu hambere ni muri bone. So this we call it predirection. Cyangwa se umuntu ufite let's say breast cancer ijya mu bihaha ijya mu mwijima ikajya mu bwonko. Ntabwo wayibona wenda ngo ajye ku gifu ndetse ya exceptional case twavuzeho kare, ntabwo yajya muri pancreas, ntabwo ukunda kujya. So abantu bakeka ko hari factors zikurura igahita imenya ngo ahangaha ninjyayo nzakirwa neza. Kwigezeyo murabibona igomba guhabwa izindi blood vessels ziyifasha kugira ngo ibeho. So this is how metastasis is done. Ikibazo kindi wagarukaho ni iki nuko mu gihe rero ukoze histology ukareba molecular aspect of this primary tumor no muvura, these they can die But this one, which has metastasized, will not die because they have acquired new antigens or new features, which will make them evade the immune system. Ibi byose bigomba kubafasha kumva uburyo wenda kuvura kanseri bigora, nubwo tuyivura dukwije molecular aspects zayo. Mu gihe bafashe sample ahangaha habanza, molecular aspects ziri hano ntabwo ziri ari zimwe n'izo muri metastatic. No kuri histology turabibona. Umuntu afite breast cancer akagira metastasis in lymph node. Iyo byombi bakuyemo histologically ntabwo zibazi. Inshuro nyinshi ntabwo zibazi. Tuhita tubona ibi bya chronic subcrones bita tubwira. Ntabwo ziba zisa niba imwe yakoraga tubules indi usanga wenda ari solid. Bivuga ko wenda hajemo subcrone muri primary tumor ikora tubule aba ariyo ishobora kuinfiltratinga ikora na metastasis. But the implication is that to treat this cancer becomes difficult because of all these things I mentioned. So mutations zigenda ziza ari nshasha. You can target mutations here and treat, but the mutations here you don't know. So that's why cancer is difficult to treat. Ariko kuri cancers zigumana the same morphological and molecular features. Hari nubwo bibaho ko utanamuhaye imiti ubaga kanseri, the primary cancer niza na metastase aho zagiye zose zigeta zishonga, ariko ni gace biba ariko biba. So we have then carcinogens, if no question so far. So as I said, the molecular basis of cancers will come back on it. There is a separate presentation. Even in this carcinogens, and what I'm presenting here now, byinshi muri byo abajya bambaza kubivuga muri community muri churches nkayo kandi bakabyumva abenshi batarize n'ikijyanye n'ubuvuzi, so ni ibintu byumvikana n'izi carcinogens. So I will give you attach this small presentation about them will read, but most of the things will cover here. So icyo mugomba kumenya nuko carcinogens are these agents which will cause cancers. So they are of three kinds. They are chemicals, they are radiation, and microbes. Chemicals, turazibona hari radiation, kuba bavuze ngo bombe atomique cyangwa za X-ray, ziriya machine za scanner, n'ibindi bishobora gutera kanseri turabireba, cyangwa se rays ziva ku zuba, ultraviolet rays, n'ahangaha biza. Hakaba microbes za bacteria, there are viruses, there are parasites which are known to cause or at least to be risk factors to cancers. So nazo ziraha. So you must know that there are these three classes. So another thing ntaribagirwa nuko muri mu bitabo bya kera muzamubona bavugamo nubu barabikoresha ijambo oncogenesis. So oncogenesis na carcinogenesis mu bitabo bya kera ni kimwe. That pathomechanism of development of cancer. Ariko ubu ngubu bivuze oncogenesis ushaka kuvuga the development of cancer caused by microbes. So niho iri jambo risigaye rikoreshwa gusa oncogenesis. Otherwise in general we use carcinogenesis. Hari n'abakoresha cancerogenesis. So bakari nk'ijambo wenda ry'abantu bo hanze, that those in the field, medical field must use carcinogenesis. So what are the chemical carcinogens? So they are in different categories. Hari ibyo bita initiators. So dusubiye hejuru kuri twavuga kuri mutation ya transformed cell iriya yagize initiators ni ibintu byatumye habaho the first mutation. Noneho factors which have triggered that mutated cell to undergo cell cycle is bazita promoters. So that is the difference. So initiators, they're the ones causing the very first mutation. Promoters is zatumye the mutated cell to be allowed to undergo cell mitosis, cell division. Ubu mu bitabo bimwe na bimwe bajyaho na third category ta progression. So initiation, there's initiation, promotion, and progression. But for me, progression and promotion is almost the same. So progression ni ukuvuga ngo cancer yamaze kugaragara no igituma wenda ikura cyane ikametastaza ikakora metastasis ni ibyo ngibyo bita progression. But it is not very much different from the promotion. So you understood that we have initiation and promotion. In initiation, we have now different categories. We have a direct acting carcinogens. Direct acting, it means this carcinogen enters into the body and automatically it causes cancer. An indirect acting carcinogen. These ones, when they are inside the body, they must be transformed. And therefore, metabolites zazo nizo zizatuma haba cancer. So they must be metabolized first in order to cause cancer. While this one, the direct acting ones, they don't need any transformation. Once in the body, cancer starts. So this, they include direct acting, alkylating, and activating agents. So igitangaje nyine ahangana uko inyinshi muri zo ni imiti ivura kanseri. Imiti ivura kanseri itera izindi kanseri. So you can ask yourself, why then giving such drugs? It is a matter of public health now. So if you know someone has a cancer and if not treated, the risk of dying is 100%. No bakavuga ati ariko ushobora kumuha umuti agacira noneho akagira risk wenda 20% or 10% yo kuzagira kanseri wenda mu myaka 5 iri imbere. Ese byakubuza kumuvura? Ugomba kumuha chance bwa mbere kuko ashobora kwisunika n'iyo myaka 10 kandi no nyuma y'imyaka 10 nta kiva ko azajya kansa kuko risk afite yo kugira kansa ni 10% icyo gihe. So urumva nta cyakubuza kumuvura nuko ibintu bisobanuka. So ntumuvuye arapfa byanze kuza. Uramuvuye arakize akagira risk nto yo kuzagira indi kanseri cyane. Urumva these risk ntabwo ari ibintu byo kuvuga ngo birangana ngo byose ni kimwe no. So you must treat. So iyi miti iri hano yose ubundi ikoreshwa mu kuvura cancer, corona, imwe ikoreshwa no mu bindi ariko nka merifarane ikunda gukoreshwa mu kuvura melanomas. Muri labo nakoraga doctoral studies bakunda kuyikoresha muri research cyane muri. So tuyigerageza ku maserera atandukanye ku cancer zitandukanye tere bishobora gukora. But it is carcinogenic. So they induce lymphoma and leukemias. The exact therapeutic effect by targeting and damaging DNA. So, noneho bayikora principe yayo yo kuvura niki. Ubundi mu kuvura kanseri ntagomba kumva ko ari mechanique no kuvura gusa. Ugomba kumva ni muhu uyu muti niki kiri bukorwe muri we imbere. Ku buryo hari n'ibintu napima nkamenya ko biri kumubamo. So icyambere nuko iyi miti yo bayitanze y'ubwayo itera icyo twakwita mutations muri DNA. Nicyo igamije. Ariko iyo mutation itera ikiba kigamijwe n'iki nuko ni izatera mutation umubiri will recognize that mutation and kill that cell. Niyo principle ikoreshwa hangana. So you give a drug, the drug causes a mutation, but a mutation which can be detected by the body and which can induce the apoptosis of that cell. So this is the philosophy of giving these drugs. So, as I said, we have procarcinogens or indirect acting carcinogens. They must first be transformed by akenshi niza bacteria ziba mu mubiri wacu bita flora, normal flora, nizo zizihinduramo. Ari ziba ku ruhu, ari ziba mu nda, mu gifu, mu mara, nizo zizihindura noneho true carcinogens. So they include polycyclic aromatic hydrocarbons, so they require metabolic activation, as I said. So where if painted on the skin, injected or introduced to specific organ, they will cause cancers. Izindi muri zo zikorwa mu kunywa itabi, combustion of tobacco, also produced from animal fat. So murabyumva animal fat ni handi noneho hari abantu babigisha bati ni ukwirinda ibiribwa birimo ibinure cyangwa se ibiribwa binure bitera kanseri cyane imbere y'amara ni igisobanuro ni cyangwa se itabi rikurura kanseri y'ibihaha, kanseri ya larynx, pharynx and so on. Ni iki ngiki you must understand what happens exactly. Ntabwo ari ukubivuga nk'umuturage ngo itabi ritera kanseri y'ibihaha. No, mechanism is by which mechanism? So it is because the products of the combustion of the tobacco will induce the initiation of mutations. So okay, and the problem here, this animal fat is produced in so they are produced from animal fat in the process of broiling meat and are present in smoked fish and meat. So aha niho ukunda kuvuga inyama zashiririye cyangwa zokeje n'ibiki, na aha nibyo bitera bikora izi aromatic hydrocarbons which will be transformed by our normal flora to become carcinogens. So therefore, if, for example, you know, in a family, many people will develop like a colon cancer, then you will be very careful about using these products. The third category of initiators, aromatic and amines azoles. So we have here beta-naphthylamine cause bladder cancer in workers of aniline dyes and rubber. Abantu bakora mu rubber cyane cyane bakora ibintu bimeze nk'amapine cyangwa amapine, abazikoramo bya rubber baba exposed kuri iyi product, then ishobora kubatera kanseri ya bladder. And unfortunately, azo dye, which was used in food color, ari ibiribwa abona bishashagirana bituma appetiza cyangwa ibinyobwa, ntabwo bari naturel irakurera biba bishashagirana cyangwa se itera appetite. Ni ibintu baba byakizwe. So unfortunately basanze bimwe mu bibyo bishyirwamo bitera cancer. So there are natural plant and microbial products, wavuga ngo kuki bitaza muri za microbes. So nuko ibi akenshi bigaragara muri nature cyangwa mu bintu ari already chemicals, not the plants or the microbes themselves. So we have aflatoxin B, murayizi ko ikorwa na fungus yitwa Aspergillus flavus, and this one will cause hepatocellular carcinoma, the cancer of the liver. Bavigrizofluvin also hari n'umuti witwa gutya uvura fungal infections. So but this drug also comes from fungus. You have saffron, ni products bisa n'umuhondo urabagirana bajya bashyira nko mu migati n'ibindi, ugasanga umugati n'umuhondo cyane, kandi abandi bayikoreshaga mu gukora perfume cyangwa parfum n'amasabune. Ibyo nabyo birazwi, they will cause cancers. And the betel nuts, bunomeza nk'ubunyobwa bugera ku giti kigaragara hanga, and these trees are, I don't think in Rwanda we have them, but I saw them near the ahantu hashyuha kandi ku nyanja nini nka nyanja y'Ubuhinde cyangwa Atlantika niho bikunda kugaragara ibingibi. So birazwi ko hano bazifata bakunda kugira cancer zo mu mihogo and so on. So we have a nitrosamine and amide.
On the best, what's up in this way?
Can you mute yourself? So these are formed in GI gastrointestinal tract of humans. So is nitrosamine and the amides nubwo zikorerwa hariya ntabwo ari igifu kizikora cyangwa intestines, ahubwo hari ibyo bita nitrostable amines and nitrates. Ni ibintu bishyirwa muri za pre-cooked foods ziba muri ma supermarket bita fast food and so on. So baba bashyiramo ibintu bituma bitabora ko ni ibiribwa bishobora kubora, ariko bashyiramo preservatives. So izi preservatives zitwa nitrostable amines and so on, iyo zigeze mu mubiri, they will be converted to nitrites by gut bacteria za normal flora, noneho iyo zibaye nitrite nibwo zishobora gutera cancer. So hari nabwo usanga nibwo uzareba muri public health bakavuga ati kanseri y'igifu n'urugero cyangwa y'amara ziganje mu bantu bafite urban lifestyle. So you must understand that in that context. No kubera iki urban lifestyle. Se no kuba barya these pre-cooked foods. No kubera ko wenda batagenda bakoresha imbaraga aho bagenda mu mamodoka bicara bakora akazi ko kwicara and so on. Ni ibyo byose ureba. Akenshi usanga ari ukubera imirire cyangwa n'ibintu banywa inzoga and so on. Za brochette ibyo byose biza muri izo lifestyle zisa. So byumvikana ko we should avoid as far as possible to use such things. Birumvikana you cannot die ubireba niba nta bundi buryo ufite bwo gusurvivinga, can take them, ariko kubigira umuco. I eat always pre-cooked food or fast food, ukumva ko uri umusirimu, ushobora kuzabisobanura mu myaka mike iri imbere ubwo busirimu. So that's why we have always to think about using the most natural things as far as possible, and to teach the population to do so as well. So other miscellaneous agents, we have asbestos. Ngira ngo naho ngaho kuri School of Medicine amabati bayakuye, ariko igihe cyose twahabaye no kugeza vuba aha ngaha yari amabati yari asakaye yari asbestos cyangwa fibrociment nk'uko bayita mu Gifaransa. Even in the campus of Remera babikuyeho vuba nta wenda n'umwaka umwe cyangwa amezi make ashize. Birazwi ko bitera kanseri bronchogenical nayo mu bihaha, mesothelioma, gastrointestinal cancers and so on. So concomitant cigarette smoking, nuko umuntu anywa itabi anaba mwenda mu mazi cyangwa akorera muri aya mazu, ibyo byongera risk zo kugira aya makanseri. Hari izindi nka vinyl chloride, workers, abantu bakora mu bintu bya plastike, PVC, ari amatibe atwara amazi, bakwita PVC, polyvinyl chloride. So abantu bakoramo kubera dust zibivamo bahumeka cyangwa se zibinjira mu bundi buryo, bakunze kugira hemangiosarcoma. Hemangiosarcoma, I hope you can now tell me. This is a cancer arising from blood vessels. Okay, in the liver, of course. There are arsenic, insecticide, aridrine, dieridrine. Nukureba niba izi ngizi atari muri insecticide dukoresha muri gihe ko nazagaragaye ko zitera amakanseri. Hari ibi ngibi, skin cancer and so on. So fibrocima. So these were initiators. The one is starting a mutation. Now mutation, a cell with mutation is there. What factors will cause that mutated cell to undergo mitosis and then have a fixed cancer now? Most of the time they are exogenous agent, but others can be endogenous. So exogenous in cigarette smoking, na viral infection. So if you followed very well, murabona ko cigarette yihagije gutera initiation and promotion. Byumvikana ko yubwaye irihagije gutera kanseri nta kindi kintu kije. Viral infections akenshi ni kuzisuzugura Epstein virus ntakivuze kuko abantu twese twarayandura ko ntacyo dutwaye, yego, ariko you don't know if you have a prior mutation which can be now allowed to be fixed into daughter cells. So we have also endogenous agents such as hormones. For example, estrogen in animals will promote liver tumors. Ngira ngo hangaha, let's be very practical here. Ku isi habaho ibibazo byinshi, hari ibivuga n'ibitavuga. Hari ni wenda n'ibivuga ntibyumvikane cyangwa ntihabwe agaciro kubera impamvu nyinshi. So estrogen, urabona ko hano byanditsemo ko itera liver tumors in animals, ntabwo they didn't precise humans. Ariko ikibazo gihari ni iki? Hari research project nyinshi zagiye zigaragaza ko estrogen, cyane cyane iyo umuntu yafashe iva hanze, no kuvuga ko ari umuntu ubiri bikoreye, ishobora gutera kanseri, kongera ibyawe kugira kanseri y'ibere, ovary and so on. Ibyo kandi evidence yabyo nuko nkuko nabivuze kare, uzasanga umukobwa atinze gushaka cyangwa wabyaye bitinze cyangwa wabyaye nta yonse, aba afite risk nyinshi zo kugira breast cancer. Ibyo ni ibintu biba observed ahantu henshi. So ubu iyo barebye rero baravuga ati nta kindi aho kiri cyane ni expose, ni uko yagize exposure nini kuri estrogen. Ikindi ni ukuvuga umukobwa wagiye muri menarche ni imihango ya mbere akiri gato cyane, cyangwa uwagize menopause, ni ukuvuga gucura atinze cyane. Umvikana niba agiye menopause, niba yaba ari imyaka 55 cyangwa 52, menarche wenda bakaba bagiye mu myaka 13, hakaba yagiye ku myaka 9, yagize exposure nini kuri estrogen. Uwe estrogen ikorwa igihe cyose, umuntu yaba atwite n'iyo adatwite, ariko iyo atwite iragabanuka hakazamuka progesterone which is protective. So ibyo rero byagiye bituma abantu benshi bemeza ko muri research ko estrogen itera ibyago by'iyi kanseri zitandukanye nk'izo navuze. Ariko kubera inyungu wenda za inganda zikorera nk'ira miti yo kuringaniza imyaro, muzi ko harimo ibifite estrogen, hari n'igira progesterone, nk'inshinge ziri progesterone, ibinini harimo progesterone na estrogen, ariko estrogen ni nyinshi. So abayikorera imiti baba bati rero ubwo ni ukutubuza isoko, so bagaha abantu amafaranga, the big professors bandi bakomeye cyane bavuga ari kwijyana ku isi, nabo bakavuga bati twebwe natwe twakoze research twasanze ahubwo estrogen iri protective. So when you read this, of course ntabwo ari wowe wakoze research, uba wasomye na hariyo ugasoma ukanaliza niba warize neza uburyo research zikorwa, ukanaliza methods, ugasanga bose ibyo badekeva bakoze ni ibintu binyuze mu mucyo, uko bavuga, uko babyandika, ntubuze ko ariko babikoze ko. Icyo gihe confusion ibitiza ndemera ibyande ndeka ibyande. So as doctors we will always have to think about this. So niyo mpamvu ahangaha utabona bayivugamo, ndetse igitabo cya WHO ngo turi bubigereho, icyo kibivuga nkanga kugeza kuri breast, baravuga ngo hari well established risk factors kuri breast cancer. Mo bajya kuri estrogen bakayishyira mu bintu biri muri debate. Urumva banga kwipronounce avuga ati no, estrogen is protective, cyangwa ngo is pro cancer, so they don't pronounce themselves, banga kugira inyungu na zimwe babangamiye. But at the individual level then, me practically here muri district hospital, what I was doing, iyo umuntu yazaga nareba history ye, kumubaza mu muryango yanyu nta abantu barwaye ibyimba by'ibere, so wasanga bihari. Icyo gihe ushobora kumenya ko hari familial cancer syndrome of breast, ovary and so on. Icyo gihe ukamubwira ati dore ukuntu bimeze nk'uko ndi kwisobanura uko nabibabwiye. None hari uburyo bwo gukoresha ibinini birimo estrogen, hari uburyo bwo gukoresha inshinge za progesterone, hari uburyo wenda bw'agapira nabira bashyira mu kaboko ni progesterone. Nkamubwira avantage na desavantage ya buri cyose agahitamo, ariko namusobanuriye. Ariko kuvuga ngo nahitamo ibinini icyo nyine yahisemo, agomba guhitamo ari informed, cyane cyane nk'ufite risk, ukamugira inama ati nkurikije ku muryango wawe, ko birahari. Hari abantu banyigishije no muri postgraduate yishe hematopathology, yari afite umudamu we yishwe na kanseri y'ibere, mukuru w'umudamu yarishwe nayo, akabaho na mama wabo yishwe nayo. Ubwo rero bikana umukobwa wese uzavuka afite risk yo kugira breast cancer. Kumwe expose ro ku muti cyangwa ku kintu kiri debatable, na conscience yawe ntabwo ibigufashije, ntabwo ibitranquille. So you will always have to judge. Simvuga ko iyo miti idakora akazi cyangwa se ko itera kanseri exactly, ariko niba iri debatable kandi hari abantu bari at high risk of developing a breast cancer, don't accelerate their cancers. Make sure protect them as far as possible. Otherwise hari benshi bafata ibinini bigeze izindi ngaruka. So nyango icyo ndagisobanuye. So there are many things behind the curtain. So the prolonged use of diethylstilbestrol, uyu ni umuti wakoreshwaga n'abadamu kera, ngira ngo bawukoreshaga batwite, n'umudamu atwite agira vomiting, nausea and so on. n'ibindi bibazo cyangwa se mwe akagera risk yo indi gashaka kuvoma uyu muti rero barawukoreshaga mu myaka y'ibihumbi 1970 none baje gusanga abadamu bawufasha nyuma iyo umudamu yawufataga ari kiri gato cyane yageraga muri post menopause akagera endometrial carcinoma cyangwa se yaba yarawufasha twita akana k'agakobwa kazavuka akura kakagira vaginal cancer. So ngira ngo umuti wahagaze mu myaka y'ibihumbi 1980 gutyo wenda benshi twavutse utagikoreshwa official but unfortunately uyu muti ngo urahari. Abantu mu Rwanda I don't know ariko hari ahantu narindi bati no hano abakobwa barayikoresha bakuramo inda and so on. So bakora imiti harabikira ariko niyo risk yayo. But otherwise it was removed from the market. So the increased intake of dietary fat causes increased incidence of bile duct, which increases the risk of colon cancer. Ngira ngo by'ibinure fatty foods or diet and so on. Ngo nta ubitindaho umuntu utarayeze mu ishuri arabivuga ibingi. But you must understand the pathomechanism as explained here. So, any question before we go to radiation? Nyango byo ni shaka kuba ari slide imwe cyangwa ebyiri and na microbes then tugasoza this part. So, radiation, they include ultraviolet rays ziva ku zuba cyane cyane, but there are machines which produce them. Hari zikoreshwa zinavura nk'abana bafite bahabaye umuhondo kuri bilirubine nyinshi kubera uburwayi cyane cyane twana tukivuka ukunda kugira babavuza ultraviolet rays ariko kuba exposed kuri igihe kinini ikibazo. So they have a free ranges so in medicine physics, but B and C they cause skin cancers ariko C ntabwo practically ntiyitera kubera ko bifatwa na ozone. But you know that people, they claim that the ozone is being destroyed by climate changes and so on. So we have ionizing radiations, X-ray, zaza arama machine afotora kwa muganga, gamma rays and the particulates, protons, neutrons, bimwe bijya muri byama bombe bakora n'ibice byose, all these they will cause cancers. So evidence babimenyeyeho ubwa mbere nuko abantu bakoze X-rays niyo bita Roentgen rays. Abantu bakoze aya maradiyo bwa mbere bavuga ati twavumbuye bayifotoraga buri kanya bakifotora barebe banezerewe bahimbawe hanyuma baza kubyishyura nyuma barwara skin cancers. So the people who works in muri za mine zikora of radioactive elements in Central Europe and the United States, they suffered from lung cancers. So radioactive elements no kuvuga za uranium n'ibindi, these ones they have developed cancers. And the most known history of survivors of atomic bomb episodes in Hiroshima and Nagasaki, they developed many cancers, leukemia, breast, colon, thyroid, lung cancers among them. Murabizi ko zatewe mu ntambara y'isi yose ariko n'ubu ingaruka ziracyahari. So probably nukuba ko n'ubwo bombe zatewe rays ziracyari aho ngaho mu kirere aho ngaho ziracyahari. Ko n'ubu hari abana bavuka badafite ukuguru cyangwa ukuboko cyangwa kubera. So therapeutic radiation is a carcinogen. So murabizi nkuko navuze ku miti n'isi radio ikoreshwa mu Rwanda muziki hari ubu Kanombe hafi bitaro bya Kanombe. It is used to irradiate cancers and kill cancer cells, but unfortunately it can cause other cancers. So microbial carcinogens nabivuze kare ko ahantu ho dukoresha ijambo oncogenesis urungubu ntabwo zigikoreshwa in general. So we have viruses, DNA viruses. As you know, viruses, it can have either DNA or RNA, not both. So the DNA oncogenic viruses, you have a human papillomavirus causing cancer of the cervix, upper respiratory tract, penis, and genital areas. You know, human papillomavirus has many subtypes, more than 100 serotypes. Zimwe ziri high risk, which cause cancer. Others are low risk. They don't cause cancer. They cause other problems. Epstein-Barr virus causes lymphoma, especially buccal lymphoma, leukemia, as I explained, I did the personal study on this in the cases from our country and others. So nasopharyngeal cancer. We have hepatitis B, hepatitis C, which cause the liver cancers, hepatocellular carcinoma. Now we have RNA oncogenic viruses, and here the most known is the human T-cell leukemia virus type 1. So ahanga bakunda gushyiramo na HIV AIDS, HIV, but HIV doesn't cause directly cancer. Its problem is that it suppresses the immunity. Kuri umuntu ugize mutation adashobora kure, they cannot repair the mutation because the immunity is too low, and the cancer cells cannot be killed. So therefore, it is indirectly induces cancers, but not a direct cause of cancers. So bacteria, Helicobacter pylori is very much known to cause stomach cancers. So no discussion about that. Fungus, we have seen aflatoxin agent, which is produced by a fungus called Aspergillus flavus, cause the liver cancers. Parasites, so malaria, it is not I can't say, it is difficult to explain. It is not as a direct cause as we can understand to lymphomas such as bacterial lymphoma. And in the study I mentioned we have done, we have also seen it induces some lymphocytic leukemias and lymphomas other than bacterial lymphoma. And they just last week we are recruiting a PhD student who will also, we will work together on that, on one aspect of lymphomas. So we had that there's interviews last week, last Tuesday morning. That's why we didn't have lectures here. So we have a schistosoma, especially hematobium, which is known to cause a chronic inflammation of the urinary bladder, and at the end it causes urinary bladder cancers. So these are just the samples. Now, this is not an exhaustive list. There are many others, but it is for you to understand that the microbes, they cause cancers. And therefore, when they give a vaccine against human papillomavirus, you can understand why. You can explain it to others. And also vaccine against hepatitis B virus and the treatment for hepatitis C virus. Because this has no vaccine, there's a treatment, which is quite available here. Hepatitis B virus, it has a vaccine, but not really a good treatment nowadays. And you know that before you start your attachment to hospitals, you must be vaccinated against this hepatitis B virus, because so when you are treating patients, so you can get infected. So better than you are immunized. So ending, we can see this part for epidemiology. So epidemiology, it just refers to now the numbers of cancers in the world. So the most common cancer in males worldwide is lung and bronchus. So 1 million new cases. So abantu miliyoni 195 bashasha b'abagabo ku mwaka. Ntabwo ari abayirwaye bose hamwe n'abari bayisanze. Aba ni ku mwaka new cases. Abagore ni breast niyo ya mbere 1,400,000. So abantu ni benshi kandi basanga hari n'abandi bari bagihari bari kuvurwa ni so. So then you can say that nyinshi ku bagabo ni lung cancer, nyinshi ku bagore ni breast cancer. Worldwide, iyo tuje mu bihugu bikennye hari ibihinduka, usanga cervix ari isa mbere ya breast. So kubapfa rero bashasha urabona ibihaha yica abantu benshi, hafi miliyoni y'undi barapfa, aba breast ni 500,000. So byumvikana ko hano hari benshi bashobora kuba bavurwa bagakira, kandi na hano mu Rwanda hari bakize. We have many survivors of cancers here in Rwanda. And you write, they help us also to indicate cancer patients, bakabigisha naboneka mufata imiti nanke twafashe twarakize and so. So when you go to developed countries, mu bihugu byateye imbere prostate ku bagabo prostate ni izi imbere ya lung. and ku bagore breast niyiza imbere n'ubundi. Ariko wajya mu bihugu bikennye mu bagabo lung niyiza imbere, bituma na global mu bagabo lung niyiza imbere, abagore breast hano niyo ije imbere, still imbere byari byahindutse. So it is breast both developing and developed countries. Ariko uje mu bihugu specific nk'ibi bya Afrika hano ni cervix iza imbere. Ari nayo mpamvu usajya batubwiraho birabura ngo this is a disease of blacks cyangwa se y'abantu bagira isuku nkeya and so on. Ibyo byose biravugwa, bavuga no kwanduranya yaza kubihisha, ariko biravugwa ari indwara y'abakene cyangwa banagira umwanda cervix. So these are statistics. So hari izindi uko zimanuka and so on. So iyi mibare igenda isohoka hafi buri myaka ibiri, bagenda updating. They update these numbers regularly after 2 years. So nko muri Amerika dore kanseri umwaka w'2011 babonye kanseri mu bagabo 422, abagore 424. Prostate was the first in men, breast was the first in women and so on. So in Rwanda, the statistics are poor. I think I have explained this last time. So nabigarukaho, ariko nka CHK muri pathology units, because of cancers are diagnosed by pathology departments, byose hari bidashoboka ko abarwayi baza baratinza bagahita bapfa ntibatugereho. Ariko muri samples zose twakira, for different reasons, 25% of them become cancers. And the most common for us is stomach, maybe because the CHK has the strongest unit of endoscopy in Rwanda. Stomach nyinshi niho ziza. The second is cervix, then breast, colorectal, lymphomas, and so on. So no kuzikurikirana. But nowadays we have what we call cancer registry. It is a project we made. It is owned by RBC. So they collect data of these cancer cases. They make sure they follow up with the patient to know what happens to them, niba bavurwa, se bitwara fata neza, n'igihe bapfuye bakabikandikwa. So ibyo byose bizatwara mu minsi zataza dufite statistics nzenda kuba nziza kurusha ubu ngubu, zenda kuba accurate ntabwo ari nziza kumutera. So zenda kuba accurate, ariko ntabwo data barazisohora ko bitangiye. So, well, there is a role in environment. Why? Because, for example, here we have Japan. So people living in Japan, the stomach cancer is the most common one. When you see Japanese immigrants who live in California, you see that stomach cancer decreases by incidence, but colon cancer increases as compared to those who remained in Japan. So the sons of Japanese immigrants, so abahungu bavutse kuba immigrants baba Japanese bagiye muri California, stomach cancer yakomeje kugabanuka, liver cancer nayo hano yongera gake, colon cancer irushaho kwiyongera, prostate cancer irushaho kwiyongera. Noneho white Californians, abazungu baho bahariya bo ukabona ko byo biri stable, imibare stable. So ahangaho byavuye kuvuga ngo ubundi birazwe ko stomach cancer yiganje cyane mu bihugu by'Iburasirazuba bw'isi muri Japan, ariko ukabona ko iyo umuntu ahavuye akajya muri Amerika, risk yo kugira gastric cancer iragabanuka. Ariko niba agiye muri Amerika kwa Nyamerika bagira risk of colon cancer izamutse, nawe atangira kugira risk yo hejuru ya colon cancer. So izi trends na hano mu Rwanda hari ibyo tubona, nka colorectal cancer it was obvious yiganje mu mujyi i Kigali. Wareba tumors nk'izifata kidneys ndetse na stenoblastoma ifata amaso yiganje muri Western Province. Leukemias, lymphomas ziganje Burasirazuba. And everything may have an explanation. Lymphomas na leukemia, because they are related to malaria somehow, uyisanga yiganje Burasirazuba n'Amajyepfo kuko n'undi niho malaria yiganje. Colorectal carcinoma yiganje mu mujyi Kigali kuko n'undi niwe afite urban lifestyle, kandi bazwa urban lifestyle it is a factor to colorectal cancer. What we don't know is why retinoblastoma and the Williams tumors they are most in the Western province. So iyo mibare mvuga ni studies twaje dukorana n'abanyeshuri batandukanye muri pathology na public health, dusanga tubona iyo trend. So it is a trend now. The next is to know why. Mbere u Rwanda tugifite laboratoire imwe ya pathology i Butare, byagaragara ko cancers nyinshi zavaga iburengerazuba cyane cyane cyangwa. So many people have tried to explain, but until now no straightforward study has been done to explain the reason. So we can discuss some risk factors for selected types of cancer. Maybe we discuss this afternoon. So kandi n'ibintu mushobora gusoma nta kigoye kirimo. As I said, ibi byinshi I presented here, except the molecular basis of cancers. Ibindi byose I presented to people in the churches and the communities who are not in healthcare field and understand. So you can read and understand. But we will come back to this at 2 p.m. and then we go to the molecular basis of cancers in details. Okay, if you have a question, you can ask it. Otherwise, then we pause and resume at 2 p.m.